Hey, what's up, everybody? Welcome back to the channel. If you're new here, my name is Naman. I made this channel so that I can go through all the milestone review sheets and overall just make your MCAT studying a lot easier and more effective. A couple of years ago, I got 100 percentile on the MCAT, and these milestone review sheets, which was created by a Redditor named Milesdown, was really helpful for my studying and overall just comprehending everything. There are these really beautiful sheets that were made based on the Kaplan review books as well as some MCAT practice exams that he took. So it was really helpful for me. And so I just want to pass it along and hopefully it'll be helpful for you. I also just wanted to say thanks so much for your emails, comments, DMs, everything. Sorry it's taken me so long to get this out. Things have been a little crazy with med school and all the busyness there, but I am using my winter holidays to try to finish off these videos. So hopefully you can pay me back by hitting the subscribe. I'd really appreciate it. And yeah, without further ado, let's go into all the biochemistry that you need to know for the MCAT. Okay, one of the most fundamental components of biochemistry is amino acids. Amino acids are basically this molecule that has four groups that's attached to that central carbon. And so this is the central carbon over here. And you can see that there's four groups. One is a hydrogen, one is the amino group, one is the carboxylic acid group, and then this random R side chain. I shouldn't say random, it's actually an R group that determines the function and basically all the properties of that amino acid. And then you should know that these amino acids actually have stereochemistry. And the reason for that is because there's four different groups that are attached to this carbon. The only one that is not chiral is glycine. And when we get into all the amino acids and their structures, you should be able to tell me why glycine doesn't have stereochemistry. But just to get that out there, it's because this R group in glycine is a hydrogen. And so because there are two hydrogens that's attached to this carbon, that molecule doesn't have chirality in the center. Now, it's very interesting from an exam standpoint, from an examiner who's writing this. They love to ask about amino acids because amino acids, this R chain, they can be hydrophilic or hydrophobic or somewhere in between. You can imagine that if you have this long chain, which is made up of carbons and hydrogens, as that R chain, then it's going to be hydrophobic. But if there's charges, if it's acidic, if it's basic, then it's going to be hydrophilic. And all the ones that don't have a charge but are not these long alkyl chains, they're somewhere in the middle of those two properties. Okay. The other reason why amino acids are interesting from an exam writing perspective is because they're amphoteric molecules. And so amino acids can exist as a base or an acid. You can see here that this group and this group, they are kind of different from each other. Blood pH in yours and my blood is usually between 7.35, 7.36 to 7.45, 7.46, somewhere between that. And so you can imagine that at that pH, this carboxylic acid, it's not going to look like this. It's actually going to lose this proton, and it's going to have a negative charge. And this amino group, it's actually going to pick up a hydrogen, and it's going to be a positive it's going to have a positive charge. And so in normal blood pH in yours and mine, we have these amino acids, and if they're just living alone, they're existing as these things called Zwitter ions, which means that they can exist with this positive charge over here and a negative charge over here. And so they have these both basic and acidic properties on the opposite sides of that amino acid. Now, it's important to think about pKa for these. And so if you remember from previous sections that I've gone over, pKa is the pH at which half of the species is deprotonated. And that's what we use titration for, to figure out what is the pKa. So let me go through that with a Google image real quick. So this is an image that I used when I was studying, and it's a titration curve. And so basically what's happening here is you have this weak acid, right? And so your weak acid, and then you're measuring the pH, and you're also measuring how much of the strong base are you adding. So you keep adding more and more of the strong base, right? You can see you start with 0, then you add 10, then you add 20, then you add 30, then you add 40. And at the point where it all shoots up like this, that's over here, that's called the equivalence point. And so that's basically the moment where all that acid has been used up because it's reacted with all of this base. And so that's why this shoots up so quickly, the pH, because now you can imagine that it was acidic, it was acidic, it was acidic, and then all that acid is used up. And so now suddenly it becomes basic because we keep adding this base, but we've run out of acid. So that's called the equivalence point. At half of that, so you can imagine that there's 40 milliliters that we've used of that base. At half of that, at 20, that's where you get this point. And this point is where pH is equal to pKa. So this is actually really interesting because the titration curve tells us about this really important intrinsic property of a weak acid, which is what is, or just any acid, which is what is its pKa? What is its basically the measure of its acidity? How strong is it? And so you can look at this, which was always helpful for me to just kind of understand it, which is that we figured out what the pKa of an acid is. And so if our pH is less than that pKa, like if our pH is 1 and the pKa is 7, then it's going to be protonated because that pH of 1 means there's a lot of protons. And it's very acidic. If the pH is greater than pKa, then that means that our acid or whatever compound is going to be deprotonated. If pH is equal to pKa, that's that situation where we got there, which is where half of that acid has been used up. And so half of it is going to be in the protonated form, and half of it is going to be in deprotonated form. Now, there's another really important concept, which is PI, and it's not going to be tested directly. Like, they're not probably, I've never seen a question where they've asked directly, like, what's the PI of this acid, but it's going to be asked indirectly in terms of like the concepts of PI. And so, because this has two different pKa's, there's a pKa of this component and pKa of this component, as well as the pKa of this R chain if it's acidic or basic. So we have a lot of different pKa's going on here. And so now we want to label these pKa's. And so here we label pKa1 as this carboxylic acid, we label pKa2 as this amino group, and then we label pKa3 as this R group. And eventually we'll learn what the exact values of these pKa's are, but you should probably know that this pKa is going to be less than 7 and this pKa is going to be greater than 7. Why is that? Because remember I told you that this is going to be deprotonated in blood pH and this is going to be protonated at blood pH. Okay, so now that we know that this single compound, the single amino acid has at least two different pKa's, possibly a third one, we can now introduce the concept of PI, which is isoelectric point, and that's where we can introduce like what is the pH at which this amino acid will have a charge of 0. And so basically how we can figure that out is let's ignore this side chain. Let's just say it's like hydrogen or something that's not acidic or basic. Basically what we can do is just take the pKa of this and take the pKa of this, add them up and divide by 2. So take the average of those pKa's. That'll tell us the PI. And so that'll basically tell us at this pH, we know that this amino acid is going to be neutral. And it's going to be a neutral Zwitter ion. That's basically what PI is telling us. And that's what it's saying here. PI, or isoelectric point, is the pH at which an amino acid is in Zwitter ion form. The charges cancel out to make a neutral molecule. And then remember I said, like, let's just pretend this is a hydrogen. Let's pretend like now that it's a basic or acidic side chain. If it's an acidic side chain, we're going to call this pKa3. And we're going to now take the average of this pKa3 and pKa1, the carboxylic acid, and that's going to tell us the PI. And if it's basic, then we'll just take the average of pKa3 and pKa2, the amino acid group, and that's going to tell us the PI. So that's basically how you calculate PI. All right, so I also went over this, which is titration, so that should all make sense to you. Now we're going to go to peptide bond formation and hydrolysis. So dipeptides are 2 residues of amino acids that are bonded together. Tripeptides are 3 residues that are bonded together. Oligopeptides are less than 20 residues, and polypeptides are greater than 20 residues. And if you're wondering why are we talking about amino acids so much and proteins so much in biochemistry, it's because proteins do almost like pretty much everything in the cell. It's like one of the most fundamental units of human life. Like you think about enzymes, you think about things that move things, you think about breaking down things. Proteins are really critical in a lot of different biochemical reactions, and so it's really important to understand all the different amino acids, the 20 really important ones that they want you to know, and then how they react with each other, how they fold with one another. And then there's a lot of different diseases that form when you lose function of an enzyme, when things misfold in your cell, when proteins are just not functional and they're not working properly. And so because of that, they're really fundamental in biochemistry. Forming a peptide bond happens through dehydration reactions. I'll show you an example of this. So this is what a dehydration reaction means. It's basically you have that carboxylic acid over here, you have that amino group over here. This hydrogen is more than willing to jump off this amino group, and this oxygen and hydrogen is more than willing to leave this carboxylic acid group. And so what you have is this leaving as water, H2O, right? You see two H's, one O. And then this carbon forms a bond with this nitrogen. So when that dehydration reaction happens, we get an amide bond. And then that amide bond is that bond between the carbon and the nitrogen. You should know that it's rigid because there's resonance with the lone pair electrons on that nitrogen. And so because of that, this molecule exists that bond between the carbon and nitrogen has partial double bond characteristics. And so because of that, it's rigid. It's not going to rotate that much on that bond. Okay, breaking up a peptide bond is a hydrolysis reaction. That should make sense. That basically means we're going to throw in water, and by throwing in that water molecule, we're going to be able to split up these two amino acid residues in a peptide bond. All right, now we have primary and secondary protein structure. Primary structure is exactly what you would expect, which is just a linear chain of these amino acid residues in a peptide, and they're going to be stabilized by those peptide bonds in between these amino acid residues. The amino acid sequence, you talk about it in the N, the nitrogen, to the carbon terminus. So you start, because when people are talking to each other, biochemists, they want to make sure that they're talking about like the chain in the right order, right? So you start from the side that starts with the nitrogen, and then you end with the side that ends with the carbon. So that's basically what that's saying. And then the N terminus is positively charged, and the carbon terminus is negatively charged, which we've already talked about. Now, when these local structures of neighboring amino acids start to bond together, there's hydrogen bonding that happens. And that hydrogen bonding is what causes secondary structure. And so I think a picture will make this really clear. So this is the picture that I used a lot when I was studying, which was really helpful to me. And so there's two types of secondary structure of proteins that are really important to know, which is the alpha helix and then the beta pleated sheet. So hydrogen bonding happens between electronegative atoms like oxygen, fluoride, nitrogen, and the hydrogen that's bonded to those electronegative atoms, right? And so you can imagine that there's this oxygen over here that's bonded to this carbon in the carbonyl, and this is going to be electronegative over here. And then there's this hydrogen that's bonded to the nitrogen. And because this nitrogen is electronegative, it's going to withdraw more of the electrons in this bond than this hydrogen will. So this hydrogen has a partially positive charge, and this oxygen has a partially negative charge. So what's happening is there's a hydrogen bond because of that electrostatic interaction. And so basically, when you take all these amino acids and you put them together, they're very reliably going to form these characteristics of an alpha helix or a beta pleated sheet because of the way that these amino acids are just lined up with each other so that they can allow for these electrostatic reactions between the oxygen and the hydrogen. With the beta pleated sheet, you can see the same thing, which is that there's hydrogens and oxygens that are all forming hydrogen bonds, and they form sheets instead of a helix. All right, next we can talk about proline. Proline is going to interrupt secondary structure because of its rigid cyclic structure. That's only going to make sense if you look at an image, so I'll show you that. So this is what proline looks like, and it's kind of this cyclic structure with this nitrogen over here. And you should just know that when you throw in a lot of prolines in a protein, that can disrupt that secondary structure. Okay, the other thing you'll see a lot on the MCAT, especially in a lot of these biochemistry, organic chemistry articles, is this process of denaturing. Denaturing is when a protein is losing its quaternary, tertiary, secondary structure due to breaking of non-covalent interactions. That can include like hydrogen bonds, hydrophobic interactions, dipole-dipole, like just any type of electrostatic reactions. Generally, denaturing doesn't affect covalent bonds like between cysteine and cysteine, and it wouldn't affect primary structure, so it's not like you're going to break the peptide bonds between each other. Now we have tertiary and quaternary structure. So tertiary structure is when we start to look at that R side chain. And so remember that R side chain, there's 20 really big ones that you need to know for the MCAT. And now you can imagine that, there's sulfurs, there's hydrogens, there's oxygens, there's all these, there's these hydrophobic R side chains, there's hydrophilic side chains, and so you throw that all together, and these these chains are now going to form like all these weird shapes and structures, and that's called tertiary structure because of all that interaction that's going to happen. It's a lot more complicated than the primary and secondary structure that we've been looking at so far. Hydrophobic reactions, that should make sense. All these hydrophobic like alkyl side chains or bulky side chains, they all just kind of want to be closer together, and they want to avoid like hydrophilic things. And then disulfide bonds is when you have two cysteine residues, and those are oxidized and they form a covalent bond between their thiol groups, the SH. And you might have heard cysteine, that's what they form when they form that disulfide bond. Quaternary structure is when you have multiple of these tertiary structures, multiple peptides that have gone through all this folding. Now you throw them together. So the classic example is when you're looking at hemoglobin, that's like 4 of these individual peptide chains that have gone through tertiary structure, and then you throw them together and you put like a heme inside, and that's what you end up with for that. So this is what hemoglobin looks like. You have these 2 beta chains, you have these 2 alpha chains, you have iron, and you have heme groups in the middle, and so that's what this looks like. That's the quaternary structure because you have multiple of these peptide chains. So speaking of that, you can also have conjugated proteins and prosthetic groups. So you can have things that are attached to the proteins, like you can have metal ions, you can have vitamins, lipids, carbohydrates, nucleic acids, and this is all like very complicated high-level biochemistry that you don't really need to think about for MCAT purposes. Denaturing, again, is that loss of the 3D structure. It's caused usually by heat or solute concentration, and you can see a lot of times researchers will use that process of denaturing for clinical or lab purposes to better understand that protein. Okay, so this is where we start to get into more of the functional things that I was talking about with enzymes, proteins, and why proteins are so important. So proteins, one of the major functions of proteins are enzymes. Enzymes are biological catalysts that are unchanged by the reactions they catalyze and are reusable. They don't alter the delta G or the delta H, nor the final equilibrium position, but they do change the rate of that reaction by altering the mechanism. And then they catalyze both the forward and the reverse reaction. So I remember I was always really confused about this until I talked to like my chemistry teacher in the 10th grade, I think, and he explained it to me really well, which was that we're not really changing like the thermodynamics of this equation. We're not of the reaction, right? So it's not like this the starting or the ending point are changing. It's more like we're just making this reaction easier to go through, both ways, both forward and backwards, right? Because we're decreasing the amount of energy that it's going to take to get to the products or to get to the reactants, right? Enzymes are kind of like that key thing that allows that to happen. And it's really hard to understand how important enzymes are until you look at like some of these, the time that it takes for a reaction to happen with and without an enzyme. It would basically take like some of the reactions that happen in our cells within like 0.001 seconds would take, I don't know, like decades to take if you didn't have the enzyme. So it's basically the reaction is not going to happen without the enzymes. Enzymes are really important biological catalysts, but the MCAT will test you on the fact that you know that the delta G and delta H are not changing. It's not like we're changing the initial reactants or the products. We're not changing the energy that they have. We're just changing the activation energy, right? It might have initially been like this. It's going to take a long time for us to get that much activation energy. With enzymes, it's going to decrease. So that's what that's saying. Now, exergonic reactions, they release energy. Delta G is negative. Endergonic, they require energy. I talked about all of this in chemistry, so make sure you look at that. And then we have oxidoreductases. These are redox reactions that involve the transfer of electrons. We have transferases that move a functional group from one molecule to another. Hydrolases, which are something that allows cleavage through the addition of H2O. So remember when I said that you can break a peptide bond, those are helped out by hydrolases. We have lyases that allow the cleavage without the addition of water or without the transfer of electrons. And then the reverse reaction is usually more important biologically. Why is that? Well, you can actually bond things together instead of cleaving them apart. We have isomerases that allows for the interconversion of isomers, including both constitutional isomers and stereoisomers. Not too important right now for you to know this, but in like med school, the Krebs cycle, that has like some really important biological isomerases. Then we have ligases. Ligases do what you think. It like pastes things together. It joins two large biomolecules. Lipases catalyze the hydrolysis of fats. So people who have like cystic fibrosis or sometimes pancreatic insufficiency, what you'll see is that they lack lipases because their pancreas is not like sending those lipases out as much anymore. And so what happens is that they develop like really fatty stools, and that fatty stool has vitamins A, D, E, and K. And so because of that, they're like missing a lot of the important vitamins that they need, just like to help you remember that, not that it's important for your MCAT. Kinases, they add a phosphate group. So they're a type of transferase, and usually kinases are something that'll like in the cell, it moves things faster, or it like makes more of the end product. And then phosphatases, they remove a phosphate group. They're a type of transferases. So usually kinases are activating and phosphatases are not inactivating. And then we have phosphorylase. Those are usually introducing a phosphate group into an organic molecule. So in glycolysis, in the Krebs cycle, in gluconeogenesis, these phosphorylases, you'll see them all the time. Now we have enzyme kinetics. So enzyme kinetics is again very important. So saturation kinetics is basically the idea that generally in a reaction, the more substrate you have, the faster that reaction, the more the reaction rate is going to be, the faster that reaction is going to proceed until you hit a maximum value. That's generally true. There are some reactions where the substrate doesn't affect the reaction rate, but we're talking about enzyme kinetics. And so enzymes, generally, they're going to move a reaction faster if you have more substrate. Now, the reason why I say enzyme kinetics is very important is obviously it's really important biochemistry, like how do these enzyme reactions happen, but they're going to test you for sure on your MCAT about enzyme kinetics. They love to test this stuff because it's very easy to write questions about this. So I'm going to try to spend some time and make sure I really hammered home these concepts. So I'm going to start with Michaelis-Menten kinetics because this was the easiest for me to understand instead of the other one, because the other one has like logarithms and stuff. And so let's start with this. Basically, on the y-axis, we have the reaction rate, right? Reaction rate is basically how quickly is this reaction moving. And so you can imagine with an enzyme, obviously it's going to be moving faster. We also have on the x-axis the substrate concentration. So again, remember, as we increase the substrate concentration, the reaction rate is going to increase until we hit a point, and that point is called the Vmax. At Vmax, the increase in substrate is not going to significantly increase the reaction rate. Vmax is basically that asymptote, the point where you're continuing to reach to, but you'll never reach. And so that's basically what this is. Again, the y-axis is reaction rate, x-axis is substrate concentration. The reaction rate is going to increase until it hits this point. We call that point Vmax. Okay, so basically Km is the substrate concentration at which you reach half of Vmax. So let's say we calculate Vmax. I'm going to make up random numbers, but let's say we calculate Vmax to be like 10 moles per second. I think that's the correct units, but let's just say it's 10, whatever the unit is for reaction time, right? At the reaction rate of 5, whatever per second, you can look at Km, you can look at the substrate, and that's what Km is. So now if I ask you the question, if Km is really high, does that mean it requires a lot of substrate or a little bit of substrate to reach half of Vmax? And you should tell me that if Km is high, then it takes a lot of substrate to reach half of Vmax. If Km is really low, that means that it doesn't take a lot of substrate to reach half of Vmax. And so basically, if Km is really low, what that means is that this reaction is like kind of moving like that, right? It's like, it's like the beginning, it's pretty efficient. Whereas if Km is really high, and this is like what trips a lot of people up, is because usually you think like low, low, high, high. But here, if Km is high, that means that this reaction, like it's going to take a while for you to reach that same point. Like you'll just keep increasing after that and reach this asymptote. But like this beginning part, it's going to take you a while just to get to half of Vmax. And so that's basically what Michaelis meant, and those are like the high yield points about that. So let's go through some of these points. Km, remember, is the substrate at which the enzyme runs at half of Vmax. That's what this equation is saying over here. And then Km is basically if you take, this is not that high yield for you to know, so I kind of don't want to go into it. But yeah, I guess I can go in. Okay, so this is basically the equation that I'm talking about. And this is how biochemists like to think about enzymes, substrates, and products. We first think that enzymes and substrates come together to form this enzyme substrate complex, and then that can form enzymes and products. And so what that equation was talking about is Km is equal to K2 plus K negative 1 divided by K1. And so I'll go back to that equation real quick. So here we're looking at K negative 1 plus K2 divided by K1. Basically what that's saying is that, let's just imagine that K1 is really high. Remember, that's the place where enzyme plus substrate forms that enzyme substrate complex. So you can imagine if K1 is really high, that means Km is going to be very low. And if Km is very low, that means that it doesn't take a lot of substrate to reach half of Vmax. And that intuitively makes sense. If the K1 is really high, that means that we're really efficient at converting that enzyme plus substrate to enzyme substrate complex. And so it makes sense that if we're really efficient at converting substrate to enzyme substrate complex, the Km should be high. Whereas if K negative 1 and K2 are very high, then we can imagine that the enzyme substrate complex, it's going to form enzyme and product, and it's going to form enzyme and substrate, like it's going to go backwards. And so if that's really high, then Km is going to be very high, which means it takes a lot of substrate to reach half of Vmax. So that's basically what this is saying. Now let's talk about Vmax. Vmax is the maximum rate at which an enzyme can catalyze a reaction. This is when all the enzyme active sites are saturated with substrate. So you can imagine that if we're going to add more substrate to a place where all the enzymes are saturated with substrate, we're not going to form product any faster because these enzymes are all busy with other substrates. But if these enzymes are like empty, like we have more enzymes, then the Vmax is going to increase. So I remember getting questions on practice tests where they're like, how are you going to increase Vmax? And they would ask like different questions, and then one of the answer choices would be, we can increase the number of enzymes. And that's one of the ways that you can increase Vmax, because if you're adding more enzymes, that means that there's more places for substrates to bind to. And so because of that, we can increase the number of substrates and actually increase the rate of this reaction. Okay, Michaelis-Menten equation, we talked about what Michaelis-Menten that looks like, but basically this is a way to look at what is your current rate of reaction. And so you basically take Vmax, which is the maximum possible velocity, and you multiply it by this fraction. And so if this fraction if Km is close to 0, that means that this is going to be close to 1, right? And then if this is close to 1, then Vmax is equal to V, which means that the rate is the place at which there's the maximum rate is the place at which Km is 0. So Km is 0 if basically it doesn't take any substrate to get to half of Vmax. That should make sense, right? If Km is very, very, very low, that means that like very quickly we can go up the curve and almost reach Vmax very soon without adding too much substrate. And so that's basically what this is saying. Now let's say Km is really, really high. So Km being really high, remember that the slope is going to be smaller and it's going to take a while to get to that Vmax. If Km is really high, then this is going to be closer to 0, right? And so if this is closer to 0, that means that the velocity or the rate of the reaction that we are currently at right now is closer to 0. So that's basically what all of Michaelis-Menten equation is saying. Now I wanted to look really quickly at Lineweaver-Burke plots also. This is not adding any new information. It's just an easier way to look at the information, because now this instead of being a curve, it's in a straight line. And so basically, we organize this information to say that the slope is km over v, and then the x-intercept is -1 over km, and then the y-intercept is 1 over v. And here they have some opportunities to ask about like math concepts. They might be like, oh, so this hits here at like -5 or something. But if we move this to the right, then does that mean that km is increasing or decreasing? Things like that. They try to be tricky. So if the line looked like this, is km increasing or decreasing? If the line looks like this, is km increasing or decreasing? If the line looks like this, is v max increasing or decreasing? If I don't know, if the line looks like this, then what's the new slope? So they'll ask things like that you just have to get comfortable with. But just remember, they're not really adding any new concepts in the Lineweaver-Burke. It's just because we're representing the information in a way that makes this a straight line. So that's kind of the important thing here. And then the last thing we wanted to mention is that Michaelis-Menten plot with the enzyme. That's one of the ways to represent how enzymes work. There's also the sigmoid shape that you can have, which happens when you have cooperative enzymes. And so an example of that is oxygen unloading in the blood and hemoglobin unloading in the tissues, and hemoglobin picking up oxygen from the lungs. And so you can imagine that when the hemoglobin is picking up oxygen from the lungs, you want that reaction to be, you want the hemoglobin to really want oxygen, right? Because you want it to pick up as much oxygen from the lungs as possible. But when it's going to distribute it to your tissues for, glycolysis and like all the things that tissues need oxygen for, you don't want the hemoglobin to hold on to the oxygen as tightly. You want it to let it go. And so that's an example of sigmoid shaped curves. So that's basically what that's saying. Okay, now we can go through some high yield points pretty quickly. These are mechanisms of enzyme activity. Enzymes act by stabilizing the transition state. So a substrate to go into the product, it has to become this transition state where it's kind of unstable. And so enzymes decrease the activation energy required by making that transition state more stable. And because it's making it more stable, it's easier for the substrate to become to get into that transition state and eventually become a product. And so that's why that's part of the reason why when we were looking at Km, we looked at K1 as the denominator, because that K1 is representing the transition from the substrate to the substrate enzyme transition state complex. So basically, the enzyme is going to act by stabilizing the transition state by providing a stable microenvironment or by bonding with the substrate molecule itself. And so those are kind of the ways that enzymes will make substrates more, why it'll decrease the activation energy. The active site is the place where the catalysis is happening, where basically the enzyme and the substrate are interacting with each other. Then there's two different theories about how these enzymes work. One is that it's a lock and key, and so it has to like match perfectly the enzyme and substrate for the enzyme to work. And then induced fit is like, it's not a perfect match, but they'll kind of like force fit each other through conformational changes and different like bonding and hydrogen bonding, things like that, that'll allow it to interact fully. There's cofactors and coenzymes. Cofactors are like metal cations, kind of like with hemoglobin, how I was showing you iron, that's a cofactor, very important. And there's also coenzymes. Coenzymes are organic molecules that are required by some enzymes to be more efficient with their enzymatic activity. Then we have effects of local conditions on enzymes. So you should know temperature and pH can really affect how an enzyme works. Salinity can also affect how an enzyme works. You can imagine if their temperature is really, really high, and you can have something happening to the enzyme, and you should remember what that is. It is denaturing. So you can imagine if the temperature is really high, then you might have denaturing of the enzyme, and so it's not going to work as well. If it's like way too acidic or way too basic, that might mess things up. If it's way too salty and there's too much salinity, that might mess things up as well. And now we have regulation of enzymes. So this is kind of important for like knowing how to increase or decrease the activity of enzymes. So we have feedback inhibition. Enzymes can be inhibited by a lot of the product. So if you've learned about this in your biology classes where you have like a lot of the product, whether it's in glycolysis or with that like lac operon in bacteria, where you get like negative feedback, and so that can decrease the amount of enzymatic activity that's happening because there's already so much product that's being developed that we don't need anymore. There's also reversible inhibition, competitive inhibition, uncompetitive, non-competitive, mixed inhibition. So I'm going to go through this in as simple of a way possible, and if there's any applicable places, I think I can show you an image. That's what I'll try to do. So this is basically the types of inhibition. You can have competitive inhibitors, you can have non-competitive inhibitors, and you can have uncompetitive inhibitors. So let's start with competitive and non-competitive. This is the place where a substrate normally binds. And let's say that now we have something that's competing for that active site. The active site is where the enzyme and the substrate are interacting to produce the product. Now, because we have something that's competing for that spot, we call it a competitive inhibitor. A question that I have for you is what if like before I had 5 substrate and now I have like 50 million substrates? Are these competitive inhibitors going to be as effective or not as effective? The answer should be it's not as effective because we can crowd out these competitive inhibitors. That's basically the major difference that they're going to test you on between competitive and non-competitive inhibitors. Non-competitive inhibitors inhibit this enzyme from a different site than that active site. And so the reason why that matters is because this substrate is going to bind over here. You have this non-competitive inhibitor over here, and what that results in is even if we add like 50 million of this substrate, like we add so much of the substrate, it's still like this enzyme is going to be inactivated by this inhibitor that's on a different site. So we can't really crowd this out, in the same way that we can crowd this out and compete for this spot. So that's the major difference between competitive and non-competitive inhibitors. Now we also have reversible inhibition. This is kind of straightforward. It's just you can compete with the inhibitor. You can replace the inhibitor with a compound of greater affinity to remove it using like laboratory techniques. So even though it's bound to the enzyme, you can reverse that binding. It's not permanent. And then competitive and uncompetitive we talked about. What does it do to Vmax and Km? This should make sense without having to read it. So a competitive inhibition should increase Km because it takes more substrate to reach the same half Vmax, but Vmax has not changed. It's still going, like we're still going to reach the maximum rate of this reaction as long as we add enough substrate. That's not true with uncompetitive inhibition. With uncompetitive inhibition, Vmax is going to decrease. Now, Km is really interesting because Km is going to decrease as well with uncompetitive inhibition. And the major reason for that is because Vmax is decreasing. If now there's fewer enzymes that are capable of acting at maximum capacity, then it's going to take less substrate to reach that new maximum capacity. And so that's why Km is decreasing. Non-competitive inhibition is when the inhibitor binds with equal affinity to the enzyme and to the enzyme substrate complex. So you can remember that equation where you have enzyme plus substrate becomes enzyme substrate complex. The enzyme substrate complex can now form enzyme plus product, or it can go backwards towards enzyme plus substrate. And so if there's a non-competitive inhibition, that means that the inhibitor is able to bind either the enzyme alone or is able to bind the enzyme substrate complex, and it binds both of them with equal affinity. So at this point, I'd like to differentiate between competitive, uncompetitive, and non-competitive now that we've introduced these concepts, because they're very easy to confuse and they're basically talking about very similar concepts, but the difference is it's what explains the differences between Vmax and Km for all of these. So we have an enzyme plus a substrate that forms this intermediate, which is the enzyme substrate complex. And then that forms enzyme plus product, right? And we know that that first part is reversible. And the second part, this part is reversible. This part is irreversible. We know that K1 goes this way, K2 goes this way. Then we know K -1 is the reverse way, right? So that's basically our equation to explain this, what's going on. Now, I'm going to let it erase. basically, these things are explaining at what point are we inhibiting the enzyme. Are we inhibiting it when it's an enzyme alone and it's being added to a substrate? Or are we inhibiting it where it's an enzyme substrate complex? Because depending on where the inhibitor wants to inhibit the enzyme, that's going to affect our mechanics. And that's definitely something that they're going to want to test you on, because it's like a very nice thing to test from their perspective, because they get to test you about like inhibition, they get to test you about enzymes, they get to test you about Michaelis-Menten. And so this is kind of something I want to hammer home for you again. Remember that competitive inhibition is something that binds at the active site for an enzyme, right? Uncompetitive inhibition binds at the enzyme substrate complex, right? And then non-competitive inhibition binds with equal affinity to the enzyme or the enzyme substrate complex. And then finally, mixed inhibitors are very similar to non-competitive inhibition. They're willing to inhibit at the enzyme or the enzyme substrate complex, but it's not like it's equal affinity. It's going to be higher affinity for one of the two. So that's what's different between these four things. Now we need to explain like why is the changes in Vmax and Km the way that they are. So you can imagine that when an inhibitor is inhibiting the enzyme alone, it's possible for an increase in the amount of substrate to overcrowd the competitive inhibition of the enzyme alone, and because of that, you can reach the same Vmax. So Vmax is unchanged when you have competitive inhibition. But when the inhibitor is affecting the enzyme substrate complex together, even if you increase the amount of substrate like 100-fold, it's still inhibiting the enzyme substrate complex. It's not inhibiting the enzyme alone. And so because of that, the Vmax is going to decrease, because the limiting factor is still that enzyme substrate complex. It's still being inhibited. When you have non-competitive inhibition, the Vmax is going to decrease for the same reason, because you're still inhibiting some of that enzyme substrate complex, even though now you're only partly inhibiting that and now partly inhibiting the enzyme. And then Vmax is still going to decrease in the mixed inhibition, because again, you're still having some of that inhibition of the enzyme substrate complex. Now let's talk about Km. So Km is going to increase when you have competitive inhibition, because you're inhibiting that enzyme at that binding site, and so it's going to increase the amount of substrate that you need to reach half of Vmax. Km is going to decrease in uncompetitive inhibition, because now it's going to take less of the substrate to reach half of Vmax. Remember, Vmax has decreased, so it's going to take less substrate to reach half Vmax. In non-competitive, remember, there's equal affinity to the enzyme and the enzyme substrate complex. So in competitive, Km is increasing, in uncompetitive, Km is decreasing. And so when there's equal affinity to both of those types of inhibition, Km is not changing. And then finally, mixed inhibition, it depends on which one it prefers. Does it prefer more of uncompetitive or more of competitive? And depending on that, that's how Km is going to respond. So remember, Vm is decreasing, but Km might increase or it might decrease depending on whether it prefers to inhibit the enzyme alone or the enzyme substrate complex together. All right, so that's, again, very high yield. Try to like rewind and make sure that all makes sense. Either that or just like make sure you like use ChatGPT or Google or something and try to understand this, because it's really a lot to get your head wrapped around. And I remember not understanding this in biochemistry class, and it took me like multiple readings of this for MCAT to actually understand this. So next we have irreversible inhibition, and that should just make sense after all that confusing stuff. Irreversible inhibition is just altering the enzyme in a way that it's not available any longer for enzymatic activity. Then we have suicide inhibitors. These are substrate analogs that bind irreversibly to the active site via covalent bonds. So now, again, no longer is that substrate binding site available for substrates to bind. Then we have allosteric effectors. Those bind at allosteric sites, and they induce a change in the conformation of the enzyme so that a substrate can no longer bind to the active site. It does display cooperativity, so it doesn't obey Michaelis-Menten's kinetics exactly, And so positive effectors, they'll exert a positive effect and increase enzymatic activity. Negative effectors will exert negative effect and decrease activity. So they're not like exactly inhibitors. It kind of just depends on the circumstances and the situation and whether it's a positive or negative effector. Okay, now we have homotropic effectors. These are allosteric regulators that's also the substrate. So again, remember that when I was talking about oxygen and hemoglobin, and remember that at certain conditions, oxygen really wants to bind to hemoglobin, and at certain conditions, oxygen really wants to leave hemoglobin. That's an example of a homotropic effector. It's where the substrate is also regulating the activity of the enzyme. Heterotropic effector is where the regulator molecule is different from the substrate. And then we have phosphorylation. Phosphorylation is basically when you're phosphorylating something for catabolism, you're activating it. And when you're phosphorylating something for anabolism, you're inactivating it. And so what that means is that when you're phosphorylating things, that means that you like to break things down inside cells. That's kind of catabolism is usually breaking things down, and anabolism is usually building things up. If you think about, like, the way that I remember this is that people in the gym take anabolic steroids, and that's to build their muscles up. Glycosylation is just covalent modification with carbohydrates. You'll see this in like different types of proteins. They'll get glycosylated. All right, I took a little break to eat, but I'm back and still going. So we talked about glycosylation, which is covalent modification with carbohydrates, and then we're talking about zymogens. Zymogens are precursors to enzymes. They are basically secreted in this inactive state, and then you activate them by cleaving them. And this is not too important for MCAT, but once you start med school, you'll learn about the intestine and the stomach and all these zymogens that are released by the pancreas and by different organs in your gut. And then they get cleaved usually by trypsin or by enterokinase, and then they become active enzymes that are able to digest the food that you're eating. Again, not that important for right now, but will come up in a few years. And then we have this reaction scheme that I went through. So again, I told you that the way that Michaelis-Menten understands enzymatic activity is that they have an enzyme and a substrate. They combine the enzyme and substrate into this transitional state, which is the enzyme-substrate complex, and that's how we end up with the enzyme plus the product. And so what this middle step is doing, the enzyme-substrate complex, is it's reducing the amount of activation energy it takes to go from substrate to product, because ideally it's making this transition state, like this is not as high energy as it would be if it was just like the substrate's transitional state alone. And then the basically what it's saying here is that the reason why we don't have like a K-2 is because we assume that almost none of the product reverts back to the enzyme substrate complex. Once it's an enzyme substrate complex, that's when it's able to go almost 100% to enzyme plus product. So that's what that's saying. All right, let's keep going. We're doing great. If you're still following along, just take a deep breath, because I know it's a lot of information to throw at you, but you're going to get through this. You're doing great. Let's just keep going. So we have cellular function, and this is what I was talking about before. So I've kind of talked about why proteins are so important in our cell. They basically, if you can think of anything that your cell has to do, usually it's a protein that's allowing it to happen. The exception is like DNA, RNA stuff that's storing information for genes to form. But like most things in your cells are guided because of proteins. So structural proteins compose the cytoskeleton, anchoring proteins. They make up much of the extracellular matrix. The most common one is collagen, elastin, keratin, actin, tubulin. They're usually fibrous in nature. In med school, you'll learn about these structural proteins a little bit more, and about different autosomal dominant or autosomal recessive diseases that happen because of people who are not able to produce collagen, for example, or elastin. But right now, just know that proteins can be structural. They can also be motor. Motor proteins have one or more head that's capable of force generation through a conformational change. And before you move through this, you should probably know like one of the really important motor proteins, what its function is, which is, muscle movement, right? If you want to move a muscle, you're going to require proteins, and that's why proteins are so important for people who are trying to build muscles. They have catalytic activity, acting as ATPases to power movement. And common application include muscle contraction, vesicle movement within cells, cell motility. Examples: myosin, kinesin, dynein. I'd recommend you check out the biology video that's on my channel, or just get through the biology sheets by yourself, because that goes through the whole muscle model a little better. Binding proteins is basically they bind a specific substrate and they're able to sequester it in the body or hold it at a steady state. And then we have cell adhesion molecules. These are a little bit more important. You might see one question or two questions on it. Cadherins are calcium dependent. I remember that because, the calcium cad dependent glycoproteins that hold similar cells together. We also have integrins. Integrins are the ones that allow cells to adhere to proteins in the extracellular matrix. And then selectins allows cells to adhere to carbohydrates on the surface of the other cells and are more commonly used in the immune system. So this is a good image to just show you. It's from a research article, and there's adherents, there's tight junctions, there's desmosomes, and again, I go through this better in the biology video, but you can just see that there's multiple types of these junctions that connect cells, and they have different levels of strength and different functions that they do. Some that allow medium strength between the cells, some that hold them really tightly together, like, as you can imagine, the tight junction. And proteins are just really important for these cell adhesion molecules, for these interactions. Okay, and then the last thing that was on the sheet was integrins. And I remember when I was going through these sheets a couple years ago, I was like, what are integrins? And this is not too important for you to know right now, but basically in med school you learn about a disease called CD18 deficiency, which is kind of a deficiency of the integrin. And what that means is that integrins are, you can see these yellow triangles, that's what these are, integrins. And usually they're on immune cells like neutrophils. And so this neutrophil is trying to go to a place that there's an infection. And it's because of these integrins that this is able to bind to the endothelial, like the blood cells, or the epithelium lining of the blood vessel. And because of that, it's able to stick. And we call this, rolling, activation, adhesion, and then finally it's able to transmigrate through there and address the infection that's going on. And so that's what basically one of the huge important relevant things biochemically integrins do. Okay, and then we also have antibodies. Antibodies are immunoglobulins. They mean the same thing. And then antibodies are proteins. They're used by the immune system to target a specific antigen, which may be a protein on the surface of a pathogen or a toxin, and then there's this variable region that's responsible for antigen binding. So this is an antibody. It's obviously really important in medicine, and you'll learn a lot more about antibodies in the first few months of med school, but fundamentally there's this variable region, right? These are antigen binding sites. This is a light chain, this is the heavy chain. Heavy chain is longer, so it kind of makes sense. And you should just know that antibodies play a very, very important role in our immune system, in adaptive immunity, and they're kind of like this really huge agent that our bodies use to respond to infections and respond to toxins and things like that. Okay, now let's talk about biosignaling. So there's ion channels, there's enzyme-linked receptors, there's GPCR, or G protein-coupled receptors. Basically, these are talking about when you have like a hormone or just a molecule that you want to get inside the cell, or you want to move something outside the cell, there's basically signaling that happens. And so if I send the body epinephrine, like my brain, or let's say my brain sends like dopamine, or so from somewhere there's a hormone that's being sent, and my cell wants to respond to it. It has to somehow tell the cell that this is the signal that's going through. And so in this example, in GPCR and G protein coupled receptor, what's happening is that there's epinephrine, and somewhere in the body is telling the cells that we want you to respond to this epinephrine. And usually what epinephrine does, it's raising the blood pressure, it's responding to all these things, and it's generally just kind of activating your body to prime against different infections. So epinephrine is really important for that fight or flight response, for dilating the airways, for doing all those things. So we see epinephrine, it's being sent by our body, and then we have a GPCR, or G protein coupled receptor. All the different types of these things, like enzyme linked receptors, G protein coupled receptor, ion channels, they'll have different ways of sending the signal down the cell. GPCRs are generally like the most complicated one, so obviously the MCAT likes to test that one. But basically, it's not that complicated. But there's a G protein, and it's activated when this binding happens. And so because of that activation, we have this GTP on it. The G protein activates in turn an adenylate cyclase. Adenylate cyclase's job is to convert ATP to CAMP. CAMP is a secondary messenger, and you'll see like there's just tons of CAMPs that's in this that's like proliferating in the cell after this adenylate cyclase is activated to convert ATP into CAMP. Like hundreds of CAMPs are now being produced, and now that CAMP will in turn activate protein kinase A. Protein kinase A is now this kind of this thing that wants to break things down, and so it's going to promote glycogen breakdown into glucose molecules, and then potentially to produce ATP and things like that. And then it's going to inhibit glycogen synthase because that builds up glycogen stores. So basically, you can imagine that epinephrine is telling our body, like, we're in this emergency situation, we need fight or flight, we need to dilate our bronchioles, we need to elevate the heart rate, we need to make the blood vessels a little bit wider. So it's activating it. And so now the cell needs to respond to that epinephrine by going through the secondary cascade and then activating the breakdown of glycogen to fuel the body's ability to respond to that epinephrine. So I hope that kind of makes sense. Well, now that you have like that basic framework, let's just go through this. Ion channels, they can be used for regulating ions in and out of the cell. And if you've watched the biology video, then you know that one of the really important ion channels is in our brain with the with neurons, and you know that sodium, potassium, calcium, even magnesium, all of these ion channels are really important in allowing signals to pass or stopping signals from passing. So you have ungated channels that are always open. You have voltage gated channels that open within a range of membrane potentials. And then you have that ligand gated channel, which means that there's a substance like a hormone or neurotransmitter that's going to activate this ion channel to open. We also have enzyme linked receptors, which is that it participates in cell signaling through like extracellular ligand binding. So basically the signal binds to an enzyme, and then the enzyme allows for secondary messenger cascades. And then we have GPCRs, G protein coupled receptors, which I've gone through, that has the alpha, beta, and gamma unit. And then basically the alpha and beta unit dissociate from the gamma unit, and that's how this cascade occurs. Okay, let's keep going. Now we have protein isolation. So protein isolation, here we're going to be talking about a lot of different laboratory techniques, and if you've ever worked in a biochemistry laboratory, you might have done some of these techniques. And usually this is how we separate different molecules, how we figure out what we're working with, and they're just kind of important in laboratory settings. The reason why we're going through this, obviously it's in the sheets, but also because it's really easy for them to write passages about biochemistry, biochemists working in a laboratory and using one of these things. So they might ask you something like, we have this protein in a quaternary shape, and we want to examine like the amino acids that are in it. So which one of these do we use? Something like that, where they're able to ask you about which of these lab techniques we're able to use. That's kind of how this will be presented to you. So the first thing is electrophoresis. If you've been in a laboratory and you got to do gel electrophoresis, what that means is that you have a negative electrode and you have a positive electrode, and you have this gel in the middle, and then you load the gel with like DNA, or in this case proteins, on one end, and you see how far they travel. And based on the based on the negative charge or the positive charge, they might move or they might not move. And depending on how far they move, you can kind of figure out different properties about this. Generally, the idea behind this is that we want to identify what protein we're working with or what DNA we're working with, and so that's how that works. We also have page, so there's native page and SDS page, and You don't really need to know much about it, but the idea here is that we're going to either denature the proteins or don't denature the proteins, and then we're going to try to figure out what proteins we're working with based on their responses to the electrodes that we're producing. So native page is we're not going to denature the proteins, and we're just going to see what happens. The only issue with that is because we're not denaturing it, every protein has its own charge to size ratio, and so depending on that, it's going to move a certain amount, and so it's difficult to compare between proteins. Whereas in SDS page, you're going to coat the protein in like a charge, and so you're going to ignore the protein's normal charge, and then you're also going to denature the protein, and so that's what's happening in SDS page, and so it's a little bit easier to compare between the different proteins. All right, next let's talk about isoelectric focusing. This is just separating proteins based on isoelectric points. So the protein is just going to migrate toward an electrode until it reaches a region of the gel where the pH matches the pI of that protein. Next, we have chromatography. Chromatography separates the protein mixtures based on their affinity for either a stationary phase or a mobile phase, and you've done chromatography in one of your lab classes for sure. This is a really popular technique. So this is paper chromatography. You can see that there's a strip of filter paper. You've probably done this before. So you place the strip of filter paper in a little bit of water, but you don't let it pass this line. And then you can see that these colors are going to move towards the water depending on their affinity towards that. And so you can like separate things based on their affinity for either that stationary phase or the mobile phase. Now we have different types of chromatography, so you can use column, you can use ion exchange, you can use size exclusion, you can use affinity, and as I'm going through this, I just want you to remember that these are all just based on how much affinity it has for one phase versus another phase. So this is column chromatography, and it's important to know that the mobile phase is the nonpolar compounds, and then the stationary phase is the polar affinity. So basically it's the same thing as the different types of chromatography, but based on the affinity to either the mobile phase or the stationary phase, you will have separation of these compounds. And so the high affinity analyte is going to be closer to that mobile phase, low affinity analyte is going to be closer to the bottom. And so then you're going to slowly, drop by drop, remove this sample, and you'll have separation of the two proteins. And this is basically the same thing, except instead now we're working with ions, and we're doing it based on its affinity to a positive or negative charge. And then we have size exclusion chromatography. This is just large versus small. So larger molecules are going to elute first because they're not trapped in that small pores. And then we have affinity chromatography. This is using a bound receptor or ligand with an eluent with a free ligand or receptor for the protein of interest. So again, these are all just ways to separate proteins from each other and isolate the protein that you're working with. It's not super high yield. It might come up in a biochem passage, but it's more important to understand like the theory behind these about why we want to separate the proteins and different properties of a protein, like the size or the charge that allows you to separate different proteins. So that's kind of the big yield. And there's YouTube videos that you can go through to really understand each of these, because I'm just kind of doing a cursory glance at them. I don't want to spend too much time because, again, I want to focus our time on really high yield concepts here. Next, we have protein analysis. So your structure of the protein can be analyzed usually through X-ray crystallography after the protein is isolated, but you can also use NMR. You might have done this in Orgo in the lab, or you just maybe have looked through NMR of different proteins. You also have amino acid sequence that can be determined using Edman degradation. You don't need to know anything about this. Concentration is determined colorimetrically, and you've definitely done this in your chemistry lab or your biology lab with like UV spec or through a color change reaction. You can use any of these assays to test for different proteins, and each of these have different advantages and disadvantages that you don't need to know about unless you want to be a chemist, which you want to be a doctor, so not applying to you. Then we have Beer-Lambert law. The absorbance is equal to the extinction coefficient. This is just a constant times the concentration times the path length in centimeters. And you probably use this Beer-Lambert law in your chemistry lab in undergrad. So that's kind of the same thing, and based on the color and the wavelength of light that's being emitted, you can figure out the concentration of the protein you're working with. Okay, we're making really good progress. So now we're going to be talking about carbohydrate classification. And so the first thing is nomenclature. You have 3 carbons, those are called trioses. 4 carbons are called tetroses. And then the really common ones that you're going to work with or see questions about is glucose, fructose, and galactose. So D and L, I think it's a little overhyped, like what you need to know about it. I never got a question on like whether something was a D or an L based on it, but you can just know that if the OH is on the right, then that's a D sugar, and if the OH is on the left, then it's a L sugar. How I remember this was left is L. And then almost all the carbohydrates are in the D configuration. Almost all the amino acids are in the L configuration. Again, I wouldn't waste too much time knowing things about this. I never really got too many questions about whether something was in D or L. I did get questions about enantiomers and diastereomers. So enantiomers, again, are stereoisomers that are non-superimposable mirror images of each other. So your hands, those are basically non-superimposable mirror images of each other. If I put a mirror in between my hands, then it would be a mirror image of each other, but I can't superimpose it. I can only like do that, but I can't like superimpose it in any way. I can't rotate it in any way to make them superimpose. So that's what enantiomers are. Diastereomers are any stereoisomer that's not an enantiomer. Again, if this is confusing to you, I really broke it down a lot more in the organic chemistry video, so just watch that. And then epimers are subtypes of diastereomers that differ at exactly 1 chiral carbon. So basically, how many of those chiral carbons are you differing your configuration at? That's what this is saying. So if it's 1, then it's an epimer. And then if it's at the anomeric carbon, then that's called an anomer. So I'm going to show you some images to explain that. So this is an image from Jack Weston's website that makes a lot of sense. So if you want to read about it, then you can read it here, and he explains it really well. But basically, I'll just look very briefly. This is an anomeric carbon, and only one of the chiral centers in this whole stereoisomer is differing at this one. And so those are called epimers. And then the anomer is usually because we're talking about these sugars, anomers is usually this carbon, which is a acetyl or a hemiacetyl carbon, and it only differs, this like anomer is a subtype of the epimer, and the stereo configuration only differs at this 1 carbon. So again, it is an epimer, but it's also an anomer because it differs at the anomeric carbon. And why does it differ? Because here it's pointing downwards, and over here it's pointing upwards. So again, epimers are basically diastereomers that differ at only one of the chiral centers, only one of the chiral carbons. If that chiral carbon is the anomeric carbon, which is like a hemiacetyl or acetyl carbon, then that is called an anomer. So it's an anomer. That anomer is also an epimer, and that epimer is also a diastereomer. So hopefully that makes sense to you. If it doesn't, then again, this is a great resource that explains it really well. Okay, let's keep talking about cyclic sugar molecules. So cyclization, usually you see these carbohydrates like glucose in this 6 carbon chain, but it can be cyclized. That's what we call it when it becomes this ring form, like over here or over here. So sugars can cyclize. I don't know how to say that, but basically they go under cyclization. You also have an anomeric carbon, so that's what I was explaining. That anomeric carbon is formed by the carbon that was in the carbonyl of the straight chain. So that's what the carbon is now, like either the acetic or the hemiacetyl carbon. That's the anomeric carbon. Then we have alpha anomers and beta anomers, and alpha is just when the OH and the CH2OH is in a trans configuration, and the beta is when the OH on the anomeric carbon is cis to the free CH2OH group. We also have Hawthorne projections. I'll show you some examples of Hawthorne projections, but basically it's a way that you can represent these monosaccharides in a 3D way, and it'll like have these lines that represent like going into the page or coming out of the page. And then we have mutar rotation. Mutar rotation is a spontaneous shift from one anomeric form to another with a straight chain form as the intermediate. So that's just explaining like how you can have these cyclic rings can have spontaneous shifts from one of the forms to another form, and that's what mutarotation means. So these are Hawthorne projections on the right, and it's a way that you can describe the cyclic chain of it. You'll a lot more of the time see these in Hawthorne projections rather than Fisher projections. Fisher projections is just another way that you can represent these linear structures. So generally Hawthorne projections is how you represented it in a cyclic form, and Fisher is how you represent it in a linear form. Okay, we have monosaccharides. These are what I was talking about before, like glucose, fructose, galactose. Glucose is the one you're going to see most commonly. They can undergo oxidation, reduction, esterification, glycoside formation. They can be oxidized into aldonic acids. So that's what this is saying. And then we can also use sugars as reducing agents. Remember, reducing agents are themselves oxidized. So sugars that can be oxidized are reducing agents. And then not super important, then you know this. Deoxy sugars, basically what that means is that the H is replacing the OH. So there's a lot of OHs on the sugar, and you can replace that with the H, and that's what you end up having with a deoxy sugar. I'm not going to mention it, but you should know like a very high yield application of deoxy. Yeah, I'll leave it at that. In the comments, you can let me know what I'm thinking about here. But then esterification, sugars react with carboxylic acids and their derivatives, forming esters. And then we have phosphorylation. So you can phosphorylate these sugars, and that's formed by transferring the phosphate group from an ATP onto the sugar. And that's a very similar reaction to esterification. And then finally, you have glycoside formation. This is the basis for building complex carbohydrates, and they require the anomeric carbon to link to another sugar. So this is what that's referring to with the glycoside formation. You basically have these two anomeric carbons, right? And they're formed, remember, the anomeric carbon from where that acetyl/hemiacetyl group used to be. So that's the anomeric carbon, and you can form a link, like a bond between them, and the bond has this oxygen from over here and over here, right? And when you form that bond, that's called a glycosidic bond. So that's what that's talking about. This is the fundamental, like glycosidic bonds are the fundamental thing that results in things like glycogen and cellulose and all those things. So it's really, really important biochemically. And so that ties in really nicely to disaccharides and polysaccharides, because disaccharides happens when you have two of these monomers that undergo glycosidic bonding, and polysaccharides is when you do that again and again and again to form a polysaccharide. Cellulose is the main structural component for plant cell walls, and starches is the main energy source form for plants. So starches include amylose, which is the really big one, and amylopectin. And then the main source of fiber in the human diet is cellulose. The reason for that is because that's like something that's not really digested really well by human beings, and so because it's not digested well, it's just adding to the osmolarity of our intestinal tract, and so it's just going to help pull things out from our intestinal tract. That's how I understand cellulose as a source of fiber in the human diet. Okay, next we have maltose, lactose, and sucrose. So maltose is glucose plus glucose, lactose is galactose plus glucose, and sucrose is glucose plus fructose. So again, that's pretty high yield, I think. Not super high yield for you to know alpha versus beta, but I guess it's helpful to know just maltose is alpha, sucrose is alpha, and then lactose is beta. That's just describing, remember, that beta is describing like a trans configuration, or sorry, the alpha is describing a trans configuration, and the beta is describing the cis configuration of the OH and the CH2OH, right? And so this is lactose is a beta glycosidic bond, and then this is an alpha glycosidic bond. So that's kind of helpful, but mainly you should know that maltose is glucose plus glucose, lactose is galactose plus glucose, and sucrose is glucose plus fructose. All right. So next we have structural lipids. So structural lipids, the characteristics of lipids is that lipids are insoluble in water and soluble in non-polar organic solvents. So basically that's just saying that they're non-polar. Phospholipids are really important in our cell membrane. They form a bilayer in our membranes, and they contain a hydrophilic head and a hydrophobic tails. That's why they form that bilayer. And the head is attached by phosphodiester linkage, and it determines the function of a phospholipid. Saturations of the fatty acid tails determine the fluidity of the membrane. So if it's really saturated, then it's going to be less fluid. That should make sense because if you're let's imagine you're like really saturated and it's really hard for it to move, it's going to have less fluid in it versus if it's unsaturated, there's a lot of like kinks in it and everything, and it's going to allow for a lot more fluid in that cell membrane. Now we have glycerophospholipids, phospholipids that contain a glycerol backbone. Sphingolipids, they contain a sphingosine backbone. Many, but not all, sphingolipids are also phospholipids with a phosphodiester bond. Not super high yield about sphingolipids or sphingomyelin, so I'm not going to beat it to death. Glycosphingolipids are also not super high yield. This will become a little bit more important in med school, where you'll have to learn like some of the like diseases that happen when people lack the sphingolipid breakdown, or they have accumulation of different sphingolipids, but not super important for MCAT purposes. Ganglioside, and also not too high yield. And then waxes, they contain long-chain fatty acids esterified to long-chain alcohols. You can use them for protection against parasites or evaporation in plants and animals. This is what a phospholipid looks like. You have that polar head that makes up the outer part of the cell membrane, and then you have the nonpolar tails, which is the inner part of the double layer cell membrane. This is what a phospholipid looks like. So you have that glycerol, you have two fatty acids, and then you have the head, which is made up of a phosphate and alcohol. And then this is the cerebroside. Again, this is not too high yield, but this is a type of glycolipid, and it's any lipid linked to a sugar that's a glycolipid. Okay. Some more like not as high yield things we'll go into here, which is signaling lipids. So terpenes, you don't really need to know this. Terpenoids, you also don't really need to know. If it comes up in a question, then just read the answer explanation about it. But you should just know that these are types of lipids that are used in signaling. And then there's steroids. Steroids are really important clinically and biologically, just because a lot of the hormones are steroids, and cholesterol is a type of steroid, so that's very important. So steroid hormones have high affinity receptors. They work at low concentration. They affect gene expression, metabolism. Cholesterol is a steroid that's important for membrane fluidity and stability, and serves as a precursor for many other molecules. So a lot of times you'll think about like cholesterol is really bad. How can we get our cholesterol down? But it's really important to know that cholesterol is actually really important for our cell membranes. They're precursors to a lot of other molecules, so they are very important clinically. Prostaglandins are also extremely important clinically. They're autocrine and paracrine signaling molecules. They regulate CAMP levels. They affect our smooth muscle contraction, body temperature, sleep-wake cycles, fevers, pains. And so there's a lot of different medicines that work either to increase prostaglandins, to decrease prostaglandins, to decrease their synthesis, that you'll learn about in med school. Vitamins A, D, E, K, you might remember me talking about these a few minutes ago. So these are all fat-soluble vitamins. Fat-soluble vitamins means that you can lose them when you're not like digesting them properly. Like if you have pancreatic insufficiency, for example, where you're not secreting enough lipases, that's an example of when you have fat-soluble insufficiencies. So vitamin A is the big like vision vitamin. It's also sometimes you use tretinoin, like dermatologists will use that, which is like a vitamin A type of medicine for acne. Vitamin D is the big one for bone formation. So vitamin D helps you to absorb calcium from the kidneys and the intestine. And so I always think about vitamin D, like that's the one in orange juice. You know, you really want to drink that. You want to make sure that your bones are strong, things like that. Vitamin E is a really big one for antioxidants. And then sometimes in the hospital you might see somebody with vitamin E deficiency who has like tingling in their fingers and toes. And then vitamin K is a really, really important one for clotting. That's kind of the major one that helps to activate a lot of your clotting factors, so that you don't, when you're like bleeding from your hand, like if you have a cut and there's like clots that'll come, or it'll be very important for stopping bleeds in the brain. So a lot of medicines that you'll give in the hospital in the future will be like either agonizing or antagonizing vitamin K activity, and so it'll help the patient's body to clot or to stop clotting. So that's very important about vitamin K. All right, let's keep going. So there's energy storage. Obviously, energy storage, you'll be thinking about adipocytes like fat cells. You'll be thinking about free fatty acids. You'll be thinking about triacylglycerides. So these are all ways that we store energy, and so lipids are very important for that purpose. So triacylglycerols are storage form of flat fatty acids. They look like this, which is a glycerol backbone plus 3 of these fatty acid chains. You can remember that that's kind of what a phospholipid looked like, except one of those fatty acid chains was actually a phosphate and alcohol. Adipocytes are animal cells that are used for storage of large triacylglycerol deposits. And you can imagine that if somebody has a lot of adipose tissue, they're going to have more adipocytes that are storing more of these large triacylglycerol deposits. And so that's why you might hear like nutritionists tell you that, oh, you should like cut down the number of triglycerides you're eating. And that's the reason why. Free fatty acids are unesterified fatty acids that travel in the bloodstream. And their salts of free fatty acids are called soaps. And so I guess with saponification, I can tell you a story about soap. So saponification is the ester hydrolysis of triacylglycerols that you're using a strong base like sodium or potassium hydroxide. So this is saponification reaction. Basically, you take a triglyceride, you have that glycerol backbone, and then you have the fatty acids. The long chain is represented by these R groups. You add it to a sodium hydroxide, and so you get this glycerol being replaced by a hydroxide, and then you have this thing, which is soap. And so why does it have soap-like properties? It's because this is negative and this is positive. And so let me show you. So this is kind of how soap works, which is that since you have that positive charge and negative charge next to each other, you'll have the hydrophilic areas on the outside, and then you have like a more hydrophobic area on the inside that are formed by the free fatty acid chains, right? And you'll have like all the grease and grub and all the like the non-polar stuff on your hands or on your arms, wherever you're putting the soap. It'll come off because it's inside this like little bubble, and so that's how soap works. So that little bubble is called a micelle that can dissolve the lipid soluble molecule in its fatty acid core, and it washes away with water because of the shell of carboxylate head groups. And so that's why when you put soap, you need to run water through it, because basically the soap, it's trapping those like fat lipid like particles on your hand, and then you run it through water, and now because it's forming this micelle with all these polar charges or like polar things around it at the heads of the micelles, it's going to just fall off with the water, and so that's how soap works. So I will say that so far, if you haven't really been paying attention, or if you haven't tried your hardest or you napped a little bit, it's okay, because the stuff we talked about before was important, but it's not as high yield for the MCAT. But moving forward, everything that I'm going to be talking about, I need you to pay attention really, really well, because all of these biochemistry things that I'm going to be talking about, like nucleic acid structure, DNA replication, repair, DNA structure, how things are organized in the cell, this page is very high yield. So nucleic acids are polymers of nucleotides, and types include deoxyribonucleic acid and ribonucleic acid. And if you were wondering what I was talking about before with that deoxy, that's what I'm talking about here, where it's really important for DNA that we have those deoxy sugars. So primary structure, this is very similar to protein structures, but primary structure is linear sequence of nucleotides. Secondary structure is the interaction between bases in the same molecule. So in DNA, the bases are held together by hydrogen bonds, and secondary structure is responsible for the shape of nucleic acids. It's not like proteins as much, where proteins, like a lot of it is tertiary structure that determines like the shape of nucleic acids, but in secondary structure, that's the hydrogen bonds here is really what's helping out with the shape of DNA. RNA second structure has 4 basic elements. There's loops, helices, bulges, and junctions. Loops include stem loops, hairpin loops, and then tetraloops, pseudoknots. That's kind of like high-level biochemistry that you don't really need to know, but I would know that these are the 4 basic elements of RNA secondary structure. Tertiary structure is the location of atoms in 3D space, and then quaternary structure is the interaction of nucleic acids with other molecules. So an example of this is chromatin interacting with histones, and I'll talk about that in this part as well. But for now, just think about like how does DNA interact with other things in the nucleus that affects its expression. So this is going through primary, secondary, tertiary, and quaternary structure. So here you can see like with DNA, you have the like string of nucleotides, and that's where you get like these G, C, G, C, U, which you'll get on practice questions. You'll get the primary structure and the list of the nucleic acids, or sorry, the list of nucleotides. Then secondary structure is how you get these helices, stem loops, pseudoknots. In tertiary structure, you start to get like different positioning of the atoms, and so that's what tertiary structure is. And quaternary structure is where you start to get it interacting with things like histones, like in DNA. Next, we have DNA structure. So DNA structure, DNA is a deoxyribonucleic acid. And so it's a macromolecule that stores genetic information in all living organisms. I think about it like it's the library, but it's basically like the genetic code that tells all the proteins, all the, like basically dictates what cell it is, what it's going to do. It has all the instructions for the cell, and so it's one of the most fundamental important units in biochemistry. A nucleoside is a 5-carbon sugar plus a nitrogenous base, but it doesn't include the phosphate group. A nucleotide is that nucleoside, but it does have 1 to 3 phosphate groups attached to it. And so nucleotides in DNA contain a deoxyribose, that's a type of sugar, and in RNA, they contain a ribose. And that's pretty much the only difference between the structure of nucleotides in DNA and nucleotides in RNA. And then I guess the other difference is that which of these nitrogenous bases are usually found in DNA versus RNA. So in DNA, the nitrogenous bases that are found are adenine, thymine, guanine, cytosine. So it's those 4. And in RNA, the uracil replaces the thymine. So you don't see thymines in RNA, you do see uracils. And so you should know that A and T hydrogen bond to each other, and G and C hydrogen bond to each other. A and T have 2 hydrogen bonds in between them, and G and C have 3 between them. And then in RNA, the U replaces the T, so the adenine is pairing with uracil via 2 hydrogen bonds. Watson and Crick are these 2 guys who created that 3D structure of DNA in this huge 3D model, where they explain that basically DNA has these alternating sugar phosphate groups that read 5 to 3 and are in these anti-parallel configuration with 2 strands that are going like in a spiral helix type of place. So we call that a double helix with anti-parallel polarity. Nitrogenous bases, purines have adenine and guanine. Those are made of 2 rings, and pyrimidines are cytosine, thymine, and uracil. Those are made of 1 ring. Chargaff's rule is just kind of, it's kind of like just an intuitive rule that the number of purines is equal to the number of pyrimidines, because if you were listening to me before, A and T bind together and G and C bind together. So obviously, if these are purines and these are pyrimidines, then and these are the ones that are like found together, then in DNA there's going to be equal number of purines and number of pyrimidines. How do you remember like which ones are purines and which ones are pyrimidines? I remember that pyrimidines is like C-U-T, cut. That's like kind of hard to remember, but the really easy one to remember is purines for me. AG is gold, so that's pure, or angels in gold, that's pure. So that's how I remember which one is purine. bDNA versus zDNA, this is not very high yield. Most DNA is bDNA that forms a right-handed helix. Denatured and reannealed, so denatured means it gets pulled apart, reannealed means it gets brought back together. So DNA replication, we're going to go through that in the appendix later on, but basically it's this very, very, very important process in our cell where you have all these different enzymes, these proteins, things that are helping us to replicate a DNA. And so there's a leading strand, there's a lagging strand, and you have to know that there's different enzymes. The very important ones is polymerase, which is the thing that's creating this new strand for us. There's primase, which is creating an RNA primer that's starting us off with DNA replication. There's topoisomerase, which is decreasing the torsional strain that's formed by all of this twisting that's going on. There's helicase, which is helping us unravel this double-stranded DNA. There's single-stranded binding proteins, which is also helping us to just keep these strands from reattaching together and also decreasing the torsion that's being formed. There's Okazaki fragments, which occurs on the lagging strand, which is formed just kind of as a function of how this DNA polymerase works. It only works in the 5-3 direction. And so because of that, on one of our two strands, which is called the lagging strand, there's going to be Okazaki fragments. And again, I'll go through this a little bit more in a different video, but for now, I want you to know that this is kind of the general process by which DNA replication works. And they're going to ask you a lot of questions about DNA replication. At least one or two will probably come up on your exam. So again, pretty important to just kind of look at this diagram and just understand it. All right, next we have DNA repair. This is going to be really important as a foundation for med school, is all of these different types of DNA and genes. So oncogenes are the ones that if there's a single mutation in it, it's going to lead to cancer. The reason for that is because these are generally just genes that function in a way that promotes activity in the cell usually. And so if you have a mutation that leads to increase in the activity of an oncogene or the activity of the end product of an oncogene, it's going to result in a lot of cellular activity, cellular proliferation, things like that can lead to cancer. Tumor suppressor genes do the opposite. So they code for proteins that are cell checkpoint inhibitors. They stop progression of cell cycle. They might decrease the amount of mitosis that's going on, things like that. And if you have two mutations in a tumor suppressor genes, then usually that'll result in some cancerous phenotype. Then we have proofreading. So DNA polymerase proofreads itself while it's working. And then the daughter strand is identified by the lack of methylation. So usually the DNA strand has methylation on it. And if one of the strands, the strand that lacks methylation, you know that that's the newly formed strand, and that's the one you can correct. So let's say that DNA polymerase is proofreading itself, and it sees oh shit, I made an error between and I had an adenine binded to a guanine. And so basically the DNA polymerase needs to figure out do I change the adenine to a cytosine, or do I fix the guanine and make it a thymine? And the way that it does that is looks at these strands and figures out which one lacks methylation. You have mismatch repair. Again, this is really high yield for med school, but these genes are really important for mismatch repair. And if you have a mutation in your mismatch repair, then that leads to some cancer phenotypes that you don't need to know about right now. But mismatch repair, the important thing for now is that you know that it occurs in the G2 phase. Nucleotide excision repair, that fixes helix deforming lesions of DNA, such as thymine dimers, and it's a cut and patch process, and it's working using excision endonucleases. And then finally, you have base excision repairs. Base excision repairs fixes non-deforming lesions of the DNA helix, like cytosine deamination, and it fixes a lot of the apyridinic, apyrimidine sites. AP endonucleases then removes the damaged sequence, which can be filled with the correct base. So I always had trouble knowing the difference between these three. And because these three show up a lot and they might test you on the differences, I'm just going to spend a little bit more time talking about these. Okay, so this is the best diagram that I could find to explain all this. So let's start with nucleotide excision repair because that was the easiest for me to understand when I was first learning this. And so a lot of times, somebody who's exposed to a lot of sun exposure on their skin, they can form thymidine dimers, which means that two adjacent thymines on the DNA strand, they can kind of covalently bond. And so that kind of creates a nick in this DNA strand. And so that's when you can't just fix one of these bases, right? You need to kind of fix that, just take out those nucleotides and replace it. And so that's what nucleotide excision repair is for. It's usually fixing those thymine dimers that are formed by sun damage. That's like kind of the very high yield thing that they'll present to you in a question. And they'll ask you what type of excision repair is it, and that's nucleotide excision repair. Then you have base excision repair. Base excision repair, generally the way that they're going to test this is they'll have somebody whose cytosine, usually cytosine, is undergoing this deamination reaction. And it's just this spontaneous process that happens in our DNA. And so when you'll have like these enzymes that will look for places where there's apurinic or apyrimidinic sites. It'll look for areas where you have that deamination reaction occurring, where that cytosine is forming a different nucleotide because it's been deaminated, and then it'll remove that messed up cytosine, and then it'll form this apurinic or apyrimidinic site, and it'll fix it, and that's what base excision repair is. So once again, base excision repair is usually for single nucleotides that are affected by spontaneous deamination reactions. And then finally you have base mismatch repair, and usually this is the one that's formed by incorrect matching of nucleotides when you're forming the new strand of DNA. And so let's say this strand is our template strand, and this says T on it, and instead of putting A, we put C. Now we need to do base mismatch repair because one of these strands we've messed up, and so that's what that means. All right, now let's talk about eukaryotic chromosome organization. All right, let's keep going with eukaryotic or chromosome organization. So we have 46 chromosomes in normal human cells that don't have like trisomy 21 or trisomy 18. 23 are from your mom, 23 are from your dad. DNA is wound around histone proteins, and those form nucleosomes. So we call the histone, that's a protein. DNA is wound around it, and that whole thing where DNA is wound around around a histone is called a nucleosome. That can be stabilized by another histone protein, and you'll see that, like, if you ever look at diagrams, you'll see that there's just DNA that's around histone 2, around histone 3, and it just keeps wrapping around these histones. And you can see that these, like, all of these structures can keep wrapping around themselves, and it'll become more and more dense. And so naturally it makes sense that the more dense this is, the more like wound up it is, the less it's going to be accessible for gene transcription. And so we call that really dense chromosomes, the really dense chromatin, we call that heterochromatin. So that's like when the DNA is really wound up very tightly, and so it's dark, dense, silent, it's not going to be expressed very well. Whereas euchromatin, it's not going to be like as wound up around the histones, and so it's going to be light, uncondensed, and expressed. Telomeres are really important genetically because they are the end of the chromosomes. They contain really high G and C content to prevent unraveling of the DNA. And during replication, telomeres are shortened, but this can be partially reversed by telomerase. So telomerase, really interestingly, you can think about cancer cells, and would you think that cancer cells upregulate or downregulate telomerases? Cancer cells will upregulate telomerases because they're going to undergo a lot of gene replication. And so because of that, all of the time, these telomeres are going to get shortened and shortened and shortened. And if it didn't have overexpression of telomerases, then eventually the DNA actual code would get interrupted and you wouldn't be able to replicate that DNA as well. And so that's why cancer cells will express telomerases to protect that DNA. Centromeres are located in the middle of chromosome, and they hold sister chromatids together. So you can remember, like, a chromatid is like that, and if you have two sister chromatids together, and you connect them at the centromere. That's what that's called. And they are separated during anaphase and mitosis. I always remember anaphase for away. The A is for away. High GC content. Remember, G and C have 3 hydrogen bonds in between them, so they're stronger than A and T. And so high GC content is used to make sure that this bond stays strong between the chromatids. Acrocentric chromosomes, those are where the centromere is located near one end of the chromosome and not the middle. So usually you'll see sister chromatids are like connected at the middle, but sometimes you'll see like that, where that's where the centromere is, and you call that an acrocentric chromosome, and that's like A for not and centric, so it's not centric. All right, let's keep going. So we have recombinant DNA, hybridization, PCR, and electrophoresis. So I'll go through this briefly, but it's pretty important for just the purposes of MCAT and Biochem and Biotech articles. So recombinant DNA is when you compose DNA of 2 different sources. And so this can be, very importantly, we use recombinant DNA in a lot of bacteria. And bacteria, if you watch the biology video or if you read the biology section, they have these things called plasmids. Plasmids are these circular pieces of DNA in bacteria that will replicate by themselves. And so biotechnologists like to take advantage of these plasmids and insert a gene of interest. So they might insert, you can imagine they'll insert like a gene that produces wool or something. And then because these plasmids will self-replicate, now you can have lots of genes of these plasmids that are expressing the gene of interest of wool. And then when you form the proteins, eventually you can end up with the product of wool. And that's just an example, but that's like recombinant DNA. That's like the biochemical application of that. Hybridization is when you join complementary base pair sequences. And then PCR is basically a way that we keep duplicating the DNA so that we get enough of our DNA of interest. So if we have one DNA strand and we want to analyze it, then we can put it through PCR, which is a process of denaturing that DNA, and then we continually replicate it using the enzymes, and then we get like thousands of these same pieces of DNA. And you might ask, why would we want to do that? And the truth is that we use PCR a ton in the hospital to diagnose a lot of viral infections and bacterial infections, and even like HIV and things like that. It's really helpful to use PCR for that purpose, where we have like a DNA sample from somebody and we use PCR and then we're able to analyze that DNA. Electrophoresis I've gone through, but that's like that gel electrophoresis. You put DNA under it in that gel, and then based on the size of it, it'll have a different negative charge, and based on that it'll go a certain amount away from the electrode, and you can figure out what's the size of the gene. An interesting application of electrophoresis is in cell apoptosis. In apoptosis, the DNA will show something called laddering, which is that it'll like split up into 180 base pair segments. And so if you put that DNA through gel electrophoresis, you'll see this really interesting pattern on a gel. And then you can figure out like, oh, these cells are undergoing apoptosis. Again, not something you have to know right now, but that's something that'll come up in like the first 2 months of med school. All right, let's keep going. Genetic code. Genetic code, the central dogma is that DNA is transcribed to RNA, which is translated into proteins. Degenerate code is that fact that multiple codons encode for a single amino acid. So you can imagine that there's like 3 things, right? There's So there's 3 places, like it could be AUG, or it could be like UGA, UAA, TAG, right? So ultimately these 3, there's going to be so many different combinations of these, because there's like 4 times 4 times 4, 64 combinations of these codes, right? But you only have like 20 really important amino acids that are going to be used. And so this code is going to be degenerate, because you're going to have some of these 3 nucleotide combinations that are expressing the same type of protein. That's what degenerate code means. Start and stop codons, this is very important. I think you should memorize this. AUG is for starting, UAA, UGA, UAG those are all for stopping. So basically what that means is that you have an mRNA transcript, and you're synthesizing a protein, and the ribosome is reading this and then and helping with the translation to produce a protein transcript, right? And how does it know when to start and when to end? It knows when to start because it sees the AUG, and then it knows when to end because it sees the UAA, UGA, or UAG. That's what this means. It's also kind of important to know that AUG is the codon for methionine, so the first amino acid is probably going to be methionine. And then the way that I remember this, there's this mnemonic that I used to use. UAA is you are away, UGA is you go away, UAG is you are gone. And so I kind of just remembered like you are away, you're gone, you go away. Those are the three that I used to remember it. Or you can just remember UAA, UGA, UAG. Okay, wobble is kind of a function of this degenerate code thing. Wobble means the third base in the codon. It allows for mutations to occur without effects in the protein. And so basically this third base of the codon is going to be like, it's not going to matter that much in terms of what amino acid is going to be put there. So that's kind of what wobbling means. Point mutation, this is going to definitely show up at least one question on it. Silent point mutations means the mutation has no effect on protein synthesis. Nonsense means that it produces a stop codon. So that's an example of this is if it was like UCA and that became UAA. That mutation is a nonsense mutation because we had a transcript and it was making like this sequence of amino acids, but because of a mutation here, now we just stopped the transcript. And then missense is that the mutation produces a codon that codes for a different amino acid, but it's not stopping the chain, it's just a different amino acid. So that's what that means. So it should make sense to you why the point mutation, if it's silent, it's usually formed, it's in the third base of the codon because of this wobble effect. That's why it happens. Frameshift mutation is a result from nucleotide addition or deletion and change the reading frame of subsequent codons. RNA is similar to DNA except for the fact that ribose sugars are there instead of deoxyribose sugars, and uracil is there instead of thymine. Also, RNA is single-stranded and DNA is double-stranded. There's three types of RNA to really remember. One is messenger RNA, which is the mRNA transcript that's created through transcription of DNA, and that is used as translation to form proteins. tRNA is the thing that brings in amino acids for the process of translation. So tRNA is really responsible for recognizing the codons on this mRNA transcript, and it'll bring the amino acid corresponding to these codons. And then finally you have rRNA. rRNA is used to make up the ribosome, and rRNA is enzymatically active, so it plays a role in that whole translation process. Transcription, again, is very important. Transcription is the process by which we take DNA and we form mRNA. So this is an image of transcription. You have a gene, and you have in the gene you have a promoter, you have exons and introns, and so basically what happens is that gene undergoes transcription, the DNA becomes RNA or mRNA, and you have to remove all the introns. That's called splicing. So you remove all these introns and then you have mRNA. So that mRNA transcript leaves the nucleus and goes into the cytoplasm, and with the help of tRNA and rRNA in the form of ribosomes, it forms this amino acid chain, right? And that happens with the help of ribosomes and tRNA. And then you get post-translational modification with all the things I told you, like glycosylation and hydroxylation and all those things. And we have like the secondary structure forms the tertiary structure, and then combines with other amino acid structures to form tertiary structure, and we finally get this protein. And this protein will either stay in the cell to do cellular activity, or it'll leave the cell to help out with extracellular activity. So that's what transcription, translation, all those things mean. Now let's look deeper into transcription. So in transcription, you have this enzyme called helicase. Helicase is responsible for unwinding the DNA double helix. You also have RNA polymerase II, which binds to this this region of TATA. It's called the Tata box, and it has a lot of T's and A's in it, and it's within the promoter region of the gene. It's about 25 base pairs upstream from the first transcribed gene, and this is where RNA polymerase II binds. So remember, transcription is DNA to RNA, DNA to mRNA. And so that's why we're going to use a RNA polymerase II instead of DNA polymerase. Now we have hnRNA. That's just a collective term for unprocessed mRNA in the nucleus. Remember, we have to remove all the introns and all those things to form mRNA. Now we have post-transcriptional modification. So this is when the eukaryotic cells, we have to take that primary transcript of RNA and convert it to mature RNA. The introns are cut out. So remember, exons exit the nucleus, they form the mRNA. Introns are spliced out. So you can remember, I just remember introns are spliced out, but you can also remember introns stay in the nucleus and they get destroyed. Exons exit the nucleus and they form the mRNA. Alternative splicing. So sometimes these introns can be cut in unique ways. Like sometimes if we have 3 introns in a gene, we can cut 2 of them, or we can cut all 3 of them, or we can cut like these 2, or we can just cut 1 of them. And so because of that, we can actually end up with different mRNA transcripts from the same gene because of these alternative splicing. And then we have, after all of that happens, then we have to add a 5' cap, and then we have to add a poly A tail at the end for that transcript, and that's how we form our stable mRNA that can leave the nucleus and go to the cytoplasm. Prokaryotic cells can increase the variability of gene products from one transcript through polycistrionic genes. What this means is that there's multiple translation sites within the gene that leads to different gene products. And so basically what that's saying is that you can take a single gene and you can end up with multiple different gene products, and that happens more often with the prokaryotic cell. So that basically this is what that's talking about. In eukaryote, a single mRNA transcript is going to form a single amino acid sequence, but in prokaryotes, it's more likely that you end up with like a single mRNA transcript, and then that's like there's going to be a ribosome that translates this and then encounters a stop codon, stops it. We get protein 1 that's a result of that, and then it sees this AUG later on in the same mRNA transcript. It goes the same ribosome, it starts here, and then it forms a second protein. So basically that's what that's saying, is that this is more likely to happen in prokaryotes, where you'll end up with multiple products of a single mRNA transcript. Okay, next let's talk about translation. Translation is the process by which we take mRNA and we make protein. So tRNA is the thing that translates the codon into the correct amino acid. So we have this mRNA transcript. It has multiple codons on it, which is these sets of 3 nucleotides, and then we have to translate it. So a tRNA comes along and binds here with its anticodon. And on the other side of the tRNA, we have an amino acid. And so when tRNAs keep doing this, we get amino acids that are here, and these form bonds between themselves. And you should remember, what type of bond is this? This is a peptide bond between these amino acids. And so when this tRNA disassociates, what we end up with is this amino acid chain, and that eventually becomes the protein. That's what's happening with translation. So ribosomes, the ribosomes that are important to know is the 70S ribosome in prokaryotes, and eukaryotes is the 80S ribosome. The prokaryotic one is made up of 2 smaller subunits of this ribosome. Those are the 30 and 50. And then in eukaryotes, it's the 60 and 40. Those are the subunits that make this up. I always remember that in eukaryotes, it's E for even, so it's 80, 60, 40. And prokaryotes is the odd one. So this is a very simple diagram explanation of what's happening in translation. So we have this huge ribosome, right? It's made up of its 2 subunits. This is a small subunit, this is the large subunit. Now we have the mRNA transcript, that's right here, and it goes from 5 to 3. And then we start with the AUG. Remember, that's the thing that codes for, and you should remember what amino acid this codes for. It codes for methionine, so that's what is right here. Okay, so we have this mRNA transcript. We see the first codon is AUG, so then this ribosome attaches here, and it starts with its P site. the P site, that there's a tRNA that comes over, and with its anticodon, which is UAC, remember UA, AUCG, this binds together using hydrogen bonding. And then on the other side of the tRNA, we have the first amino acid, which is methionine. Now what's going to happen after this is this ribosome is going to slide over this way, right? And it's going to read the next codon. So when this ribosome is sliding this way, then this tRNA is moving to the E site. So E is for exit. So what happens is that this moves this way. It's about to exit. And then we have this new guy that's arriving. So A is for arrival. And so what happens is that this guy arrives, it takes up the A site. So now we have one in the P site, we have one in the A site. This is tryptophan, right? These form a peptide link, and then it all moves over. This guy moves to the E site, this guy moves to the P site, and now we have a new thing moving to the A site. So that was a lot. Let me just go over it again. We have the mRNA transcript. There's 3 codons. We have the P site, E site, A site. A site is for arriving, E site is for exiting. The first thing is methionine. The tRNA binds at the P site. Then the ribosome it's going to first accept this new tRNA into the A site. Then everything is going to move over as the ribosome wants to continue reading. This tRNA moves this way, and then this tRNA moves to the P site, and then the new tRNA is going to go to the A site. And it's going to keep going like that. As things are exiting the E site, new things are arriving at the A site. And these guys are going to find peptide bonds. Okay, some other high yield points here is that there's the Shine-Delgarno sequence. That's where the 30S ribosome attaches, and that's where it scans for the start codon. So remember, it starts translation at the start codon, but it starts to attach at the Shine-Delgarno sequence. And then it lays N-formal methionine at that P site for the ribosome. That's what the AUG is coding for. And then in the eukaryote, the 40S ribosome attaches to the 5' cap, because remember in eukaryotes there's the 5' cap, and it scans for the start codon, and it lays down methionine in the P site of the ribosome. So remember, in prokaryotes there's the Shine-Delgarno sequence. That's what the small ribosomal unit attaches to, and in eukaryotes what it attaches to is the 5' cap. Okay, then we have elongation and termination. So elongation is what I explained that whole EPA process to. The tRNA is, the first one goes to the P site, the next one goes to the A site, and now what was in the P site moves over to the E site. What was in the A site moves over to the P site, and something new comes to the A site. And now they're just going to keep going, and whatever's in the E site leaves, the tRNA leaves, but it leaves back the amino acid, which is forming this peptide linkage with the next amino acid. And so that's how the amino acid chain grows. Now we have termination. Termination occurs when the codon in the A site is also a stop codon. And so you can imagine that now there's a tRNA that's coming, binding to this mRNA sequence. It has an anticodon, and the anticodon is binding to a codon, which is one of these 3. So now that's the signal that, okay, we need to terminate this whole process. And so then there's a release factor that places a water molecule on the polypeptide chain and releases the protein. So the protein is now finally going to leave this ribosomal mRNA, tRNA complex. Okay, we have post-translational modifications the same way we had post-transcriptional modifications. These post-translational modifications happens through chaperones. And this is done through formation of quaternary structure. We have cleavage of proteins and signal sequences, and then we have covalent additions of other biomolecules. Phosphorylation is a really big one. Carboxylation is a really big one. Glycosylation is a big one. So yeah, those are kind of the big ones, but you should just know that chaperones is really what's very important for like the whole folding of the proteins. And then we have DNA ligase. DNA ligase just fuses DNA strands together to create one strand. I think this was just misplaced, but this is all talking about translation otherwise. Okay, control of gene expression in prokaryotes. So we have the Jacob-Monod model, and explains how operons work. Not too important that you know like the name of this model, but just what operons are. Operons are inducible or repressible clusters of genes transcribed as a single mRNA. And this is, remember, for prokaryotes. So you have This is not true in eukaryotes, but in prokaryotes, you have clusters of genes that are transcribed together as a single mRNA transcript. Remember, that's how we get multiple different products of a single mRNA transcript. That's how we get multiple proteins from the single mRNA transcript. That's what's called an operon, that single cluster of genes that forms the single mRNA. Inducible systems means that under normal conditions, they're bounded to a repressor, and they're turned on when an inducer pulls the repressor off. So that's what the lac operon is. I'm sure you learned about the lac operon in your biology class. They love to test about it and teach about it. It's just kind of one of those things it's really easy to write questions about it. But inducible systems, just know that at baseline, there's a repressor on. You have to take an inducer and take that repressor off. And repressible systems means that under normal conditions, they are transcribed. And then they're turned off by a co-repressor that couples with a repressor and then binds to the complex at the operator site and turns it off. So just know, inducible systems, you have to put in effort to induce it, to turn it on. Repressible systems, you have to put in effort to stop it, to repress it. And so the lac operon is inducible, the trip operon is repressible. All right, next we have control of gene expression in eukaryotes. Transcription factors means they're basically proteins that search for a promoter and the enhancer region of the DNA. They bind to that DNA and then they recruit RNA polymerase. Transcription factors is really important, so I want to just talk about it for 1 second. Transcription factors is a heavily regulated thing in biochemistry and biology. You're going to encounter it a lot in a lot of medications you'll learn about in medical school, where you'll want to upregulate or downregulate transcription factor activity so that you're able to produce more mRNA transcripts, or you're able to decrease the number of mRNA transcripts. And so transcription factors are very important for control of gene expression. You have promoters and you have enhancers. Promoters are within 25 base pairs of the transcription site. Enhancers can kind of be like really far away, and the DNA will fold to allow for enhancement of the activity of transcription. So remember, promoters is like kind of where the polymerase is binding to start the whole polymerase of the DNA. Enhancers, they're just like, they can be pretty far away, but they're there to help increase the amount of transcription that's happening. Okay, and then modification of chromatin structure affects the ability of transcriptional enzymes to access DNA through histone acetylation or DNA methylation. So this is really, really important, and I'm going to hammer down on this for 1 second, which is that, remember when I told you that how tightly DNA is wrapped around histones affects how much transcriptional activity is going to happen? That happens through histone acetylation or DNA methylation. If we're going to methylate DNA, we're going to decrease the availability for transcription to occur. If we're going to acetylate histones, that means that we're going to increase the availability of DNA transcription to occur. So just remember acetylation. I always remember acetylation for activation. Acetylation is allowing DNA transcription to occur. So remember that. All right, so I'm going to be honest, this page, biological membranes, is not as high yield as the other pages, and it's pretty dense. So what I'm going to do is I'm going to try to show you a lot of photos so that from Google Images, so that it makes more sense. But yeah, let's get through it. So fluid mosaic model is it's basically this idea that represents the cell membrane, which is that it's a fluid mosaic model. Things are like constantly shifting and moving, and so it's not like this really hard structure to penetrate. And so it's made up of this phospholipid bilayer that you can see down here, which is that there's this phospholipid head on the outside and phospholipid head in the inside, and there's these tails in the middle, and it's a bilayer. So there's one phospholipid layer over here and another phospholipid layer over here. And you can imagine that the phospholipid head, as you remember, is hydrophilic, and the tails are hydrophobic. And so this inside is hydrophobic, the outside is hydrophilic, and these parts of the outside of the cell and the inside of the cell are are like aqueous, so they're more hydrophilic regions. Then we have lipid rafts. You don't really need to know them, but they're basically the idea that lipids can move freely in the plane of the membrane, and they can assemble into lipid rafts. So lipids can move freely in this way. And then flip bases, I've never gotten a question on this, but it's the specific membrane proteins that maintain the bidirectional transport of lipids between the layers of the phospholipid bilayer in cells. Then you have protein and carbohydrates, and they can transverse this phospholipid bilayer because they're big and they're heavy. They're going to be slow. And then you have these membrane components. So lipids, as we just mentioned, are the primary membrane component. It's both by mass and mole fraction. So lipids include triacylglycerols and fatty acids. This is the phospholipid precursors that make up a phospholipid. And then you have the glycerophospholipid, which is just replacing one of the fatty acids with a phosphate group. And that's often linked to the other hydrophilic groups. Remember, the phosphate group is hydrophilic. Then you have cholesterol. Cholesterol is really important in our cell membranes. So everybody always likes to think about like cholesterol is really bad for our diet and all those things. But cholesterol, remember, is like this really important biological component as well that contributes to our cell membranes. And so this should make sense as well, which is that increasing temperature decreases fluidity, and then decreasing temperature increases fluidity. And that's kind of counteracting what would normally happen, and that's kind of like what the cholesterol is helping out with. Okay, now you have waxes. Waxes is, again, not that important, but it's present in very small amounts, and if at all, they're most prevalent in the plants and function as waterproofing and defense. So if you've ever like had somebody who is using plant to like waterproof their jacket or something, that's like one of the components that they're using. Transmembrane proteins are things that are like within the phospholipid bilayer. They span the whole membrane and they allow things to pass through, and a lot of times they're glycoproteins. Embedded proteins are usually part of like a catalytic complex or involved in cellular communication. Remember, like there are those proteins that are involved in electron transport chain, so that's an example of embedded proteins. Membrane-associated proteins are these things that are recognition molecules or enzymes. In med school, you'll learn all about the immune system in more detail, and then there's like these membrane-associated proteins that are really important for recognition molecules, and that's what activates your whole your immune system pathway to counteract to bacteria and viruses. Carbohydrates can form protective glycoprotein coats, and they can function in cell recognition. Ligands are, you know what ligands are, we've reviewed them already, but they're extracellular ligands. They can bind to membrane receptors, and they can function as channels or enzymes and secondary messenger pathways. And you should know examples of ligands, right? Like the whole JAK-STAT pathway, and then the non-tyrosine kinase receptors, tyrosine kinase receptors. You probably heard of these things. And so extracellular ligands, they can bind to membrane receptors, and then they can activate these secondary messenger pathways that result in all these things happening within a cell. Gap junctions, tight junctions, desmosomes, and hemidesmosomes. I always like to look at pictures for all of these because it helps me remember them. But basically gap junctions, that's the easiest to remember. Gap, there's a gap, so it's allowing ions, other small molecules to transverse across cells. Then you have tight junctions. Tight junctions also make sense. It's kind of like, it's very tight, right? It's not like a gap junction. So it's preventing solutes from leaking into the space between cells via paracellular, so between cells, and it doesn't provide intracellular transport. Desmosomes and hemidesmosomes. So in med school, like I started to appreciate desmosomes and hemidesmosomes more because there's like some pretty important diseases that happen because of antibodies to desmosomes and hemidesmosomes. But for this purpose, I think it's important to just know that desmosomes bind adjacent cells using the cytoskeleton, and then hemidesmosomes are kind of similar, but instead of like binding the cells together, they're binding the epithelial cell to the basement membrane, to the surface. So hemidesmosomes, I kind of think like it forms a H type of structure. I don't know if that's like too helpful. It's more like a T, but it's attaching the cell to the basement membrane, and then the desmosomes are attaching cells together. Okay, let's look at some pictures. So I think this image over here shows pretty well what I'm talking about when I talk about the fluid mosaic model. There's just tons of things going on. You can see a protein channel that we were talking about that allows things to move. You can see cholesterol over here that's counteracting like the fluidity that normally happens with temperature. You can see like globular proteins, you can see glycoproteins, you can see integral proteins, you can see surface proteins, you can see hydrophobic tails, you can see hydrophilic tails, carbohydrates, and then you can just see this whole phospholipid bilayer that makes up the bulk of this stuff. The other thing I wanted to show you was hemidesmosomes versus desmosomes. So here I think you can see all the different things. So you see tight junctions, that's not really allowing anything to go through. You see adherent junctions, and adherent junctions are really dependent on calcium. You can see desmosomes that are with these cytoskeletons that are attaching to it, and then you can see gap junctions, and you don't really see much in the gap junction except for the fact that there's a gap. And then you can see hemidesmosomes way down here, and this is the basal lamina, so this is like attaching it to the surface or the basement membrane, the basal lamina. So here is another example of what I'm talking about. This is the basal lamina, this is the laminin, and then this is the hemidesmosomes. All right, back to this stuff. So again, not as high yield as the other stuff we've been going through, but there's concentration gradient. So all transmembrane movement is based on concentration gradient. And then the gradient can tell us whether the process is active or passive. So that should make sense, right? If there's a lot of sodium on one side and very little sodium on the other side, where do you think the sodium is gonna want to go? It's gonna want to go from high to low. But if it's going from low to high, then that tells you that that process is active. There's probably ATP involved in forcing the sodium to go against its concentration gradient. Osmotic pressure is a colligative property. It's basically the idea that if there's a lot of solutes on one side of the membrane and very little solutes on the other side, then that probably means that the side with more osmoles, it's going to suck the water. Like the water is going to want to go from the place that there's less osmoles to the place that there's more osmoles. So it's kind of like you can think of it like a sucking pressure, where the solutes are drawing water in, and this is the equation for that process. Passive transport, you know what passive transport is. It doesn't require energy. It's what I was just talking about. Simple diffusion is the form of passive transport, and it's when there's no like proteins involved. It's just small non-polar molecules that are moving across. Osmosis is that, but for water. And then facilitated diffusion is basically passive transport, but there's a transport protein that's helping things move along. So it might not be the small solutes that are moving, it might be bigger things that require these transport proteins for help. Then you have active transport that requires energy in the form of ATP. You have primary and secondary. So primary is using ATP directly to force things to move across the concentration gradient. And then secondary active transport is coupled transport. So let's say there's a lot of sodium on one side and very little sodium on the other side, and we're forcing the sodium to go from the less side to the more side against its concentration gradient. Instead of using ATP to force that process, we might couple it with another solute like chloride that's moving down its concentration gradient, so from high to low. So because chloride is going from high to low, it's allowing the sodium to go from low to high, and that's like coupled transport. You can have symport and antiport. That just means are the solutes going in the same direction or opposite directions. You'll learn eventually all about like the nephron and the kidney and how like different solutes move along the gradients at different parts of the nephron. Endocytosis and exocytosis is the method of engulfing material into cells and releasing the material out of cells. So you have pinocytosis and phagocytosis, and phagocytosis is all about ingestion of larger solid materials, and pinocytosis is smaller materials through these little vesicles. Specialized membranes are basically you have the membrane potential. So this is maintained by sodium-potassium pumps. Sodium-potassium pumps use ATP to force sodium and potassium to go against their concentration gradient. And usually the resting potential of most cells is between -40 and -80. If you think about neurons, this is where it's like very important, because maintaining that resting potential is very important for when eventually the neuron will create that action potential. Nernst equation, this is electrical potential. If you've gone through the chemistry video, you should understand all of this. And then Goldman, whatever this is, it's not important. I never got a question on it, so I wouldn't worry about it too much. I remember really stressing out about this equation. But if you really feel the need, then I would watch a short YouTube video on it from I think it was MIT. There was a YouTube video on it that explained it pretty well, but don't worry about this too much. Mitochondrial membranes, there's the outer mitochondrial membrane, inner mitochondrial membrane. The outer one is highly permeable to metabolic molecules and small proteins. The inner one is where the ETC is happening, where the citric acid cycle is producing those electrons that are used in the ETC. This does not contain cholesterol. Okay. And when I said ETC, that's electron transport chain. Okay, now we have the real bulk. So I said this page wasn't as high yield. This page is very high yield, so I would really focus on this. I'm going to try to give you some tips about understanding all this before we get into it, but the major tip I have is that reading this is not going to help as much as just looking at a lot of diagrams, a lot of videos. Like Khan Academy is really good for this stuff. Just look at diagrams, look at images, look at videos. That's going to help you a lot more than reading this stuff, because this stuff is stuff that you only remember before major exams. You have to remember this for probably your biology exams. You have to know this stuff for USMLE Step 1. You have to know this for the MCAT, but then eventually you'll forget. It's kind of the meme about remembering and forgetting the citric acid cycle and all those things. So yeah, just look at a lot of images and videos, remember it for the purpose of this exam, and then you can forget it afterwards. So let's first talk about glucose transport. So there's different types of glucose. Remember, glucose is kind of this bigger molecule, right? And so we need to have proteins that are helping to transport the glucose in and out of cells. And remember, glucose is a really important thing that we need to get into cells and out of cells, right? If you're eating sugar and let's say that you're diabetic and you're not responding to insulin, then that glucose is not getting into your cell, and that's why diabetics have such big problems. And so these transporters are really important. GLUT2 and GLUT4. So GLUT2 is found in liver. Liver is this primary place of glucose storage. And then you have the pancreatic beta islet cells, and that's used as a glucose sensor. And this has an increased Km. Remember the top bit, what I was talking about, Km. So remember that stuff. And then you have GLUT4, which is found in adipose tissue and muscle cells, and that's stimulated by insulin and has low Km. Okay, so again, just look at diagrams and images to remember this stuff. This is kind of just something that you have to memorize, not as high yield as the other stuff we'll go into in this page. Now you have glycolysis. So you should know this equation. Generally, you have glucose that forms 2 particles of pyruvate. Glucose is a 6-carbon molecule. Pyruvate is a 3-carbon molecule. So you're not losing any particles of carbon. You have NAD plus that's becoming NADH. 2 particles of NAD plus become 2 particles of NADH. You have 2 ADP plus phosphate become 2 ATPs. So why does this happen? Well, you're producing pyruvate, which is going to go on form acetyl-CoA, which is going to act in the citric acid cycle. And you're also importantly forming 2 particles of ATP. So remember, glycolysis and the Krebs cycle and the electron transport chain, this is all how your cell is using glucose from the food that you've eaten in order to produce ATP so that your cell can do work, so that it can use the energy that's created by this whole process. So glycolysis is that first step, and you have multiple important enzymes. Let's look at the appendix here. So you have all these steps that are happening in glycolysis. So you have glucose, which is the 6-carbon. Remember, it's forming pyruvate, which is 3-carbon. This is all happening in the cytoplasm. You have glucose, which is forming glucose 6-phosphate. This first step can happen either through hexokinase or glucokinase. I'm just going to double check that. Glucokinase, I think that's right. Yep, yeah, it's glucokinase and hexokinase. Those are the 2 enzymes that allow for that first step to happen. Glucokinase is found in the liver and the pancreatic beta cells, and hexokinase is found in the skeletal muscle, the heart, the adipose tissue. And glucokinase has lower affinity for glucose, so it requires higher glucose concentration, and hexokinase has a higher affinity for glucose. So even at low concentrations, it can function. Okay, now where are we producing molecules of ADP and where are we producing molecules of ATP? So it's important to know that in your first 5 steps, in order to produce like to break down your 6 carbon molecule into 2 3 carbon molecules, it's going to take 2 ATPs in order to do that. These are like activating steps, right? So you're going to take ATP, and it's going to hexokinase is going to require ATP to form this, to take this glucose and form glucose 6-phosphate. So where is that phosphate coming from? It's coming from that phosphate from the ATP. That's the first step. Now you have glucose 6-phosphate, and it's just going to isomerase. Remember we talked about in the orgo section, isomerase is just going to rearrange this. So it's not going to require any ATP. From glucose 6-phosphate, we form fructose 6-phosphate. Now, this is a really important step, phosphofructokinase. It's going to take this and it's going to add another phosphate at the 1 in the 6th position. So we had a phosphate at the 6th position. We take an ATP, we form an ADP, and that extra phosphate goes on the 1 position. Now, this is the step where we break things down. So fructose 1,6-bisphosphate now breaks down into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And these are isomerases of each other, so there's an enzyme that connects the two. But this step is very important, understanding that fructose 1,6-bisphosphate is breaking down into 2 3 carbon molecules. Now, those 3 carbon molecules are going to get phosphorylated into 1,3-bisphosphoglycerate, right? So basically all that's happening is that a phosphate is getting added to this glyceraldehyde 3-phosphate. Importantly, this step does not require ATP. You're just adding a phosphate to this to form this. Okay, now here's where we get our payoff, right? We are going to take that phosphate that we just added and add it to an ADP in order to form ATP. But remember, there's 2 of these particles, so the 1,3-bisphosphate is forming 3-phosphoglycerate. That's happening twice, so you're producing 2 ATPs. So the 2 steps that we just did of ATP investment, we just paid off right here. Now we're going to do a series of steps where we mutate this. We're just taking that phosphate group, moving it from the 3 position to this 2 position, and then there's going to be enolase, which removes the water, and you get PEP. And we're almost there. And PEP, we remove a phosphate group from there to form another ATP, and we get pyruvate. So remember, there are 2 of these 3 carbon molecules. So in return, there's 2 ATP that are formed here. These 2 ATP are basically paying off the ATP investment that we had in the first 5 steps. And then we have 2 ATP that are formed here. So finally, we end up with a pyruvate. We end up with 2 ATP molecules. That is happening through pyruvate kinase. And we didn't produce any carbon dioxide. We just produced 2 ATP, 2 pyruvate. And then we also produce 2 NADH. Remember, NADH is formed in that first step, glyceraldehyde 3-phosphate dehydrogenase. So we form 2 NADH right there. All right, so that's glycolysis. That should make sense to you. If it doesn't make sense, watch a video about this. Look at a lot of diagrams. This is very important. This is the first part of the 3 parts of breaking down glucose. So make sure you understand that. All right, so next we got to talk about pyruvate dehydrogenase. So there's a process by which we have to take the pyruvate and we have to form acetyl-CoA, because pyruvate does not participate in the Krebs cycle, but acetyl-CoA does. So there is a complex of enzymes that convert pyruvate to acetyl-CoA right before the citric acid cycle. It's stimulated by insulin and it's inhibited by acetyl-CoA. That should make sense. Acetyl-CoA should inhibit this process because if we have too much acetyl-CoA, we don't want to produce more. But insulin should stimulate this process, because if we have blood sugar and we have high blood sugar, insulin is getting released and it's telling our cells we want to take in this glucose into our cells and we want to break it down. So we take the pyruvate dehydrogenase, we take the pyruvate and then we use pyruvate dehydrogenase kinase using an ATP, and basically all right, let me start over. So basically we have pyruvate over here and we have acetyl-CoA over here. And pyruvate dehydrogenase is active over here through a series of enzymatic steps. Pyruvate dehydrogenase is going to allow us to convert pyruvate into acetyl-CoA. What's going in is an NAD+, and what's coming out is an NADH out of this process. Pyruvate is a 3-carbon molecule, and acetyl-CoA is a 2-carbon molecule. So, as a result, we're releasing 1 carbon. That carbon gets released as a carbon dioxide. Pyruvate dehydrogenase, and this part is a little bit lower yield, but pyruvate dehydrogenase over here is active. It's allowing this enzymatic process to continue. But it can also be inactive, and that happens when it's phosphorylated. What do you think phosphorylates PDH? It's PDH kinase. And what do you think removes the phosphate from PDH when it's inactive? PDH phosphatase. So that's basically what is happening over here. PDH is allowing this to go forward. Okay, so now we have acetyl-CoA. We've completed the glycolysis step, and then eventually we're going to go into what happens with acetyl-CoA over here, right? With the oxidative phosphorylation and electron transport chain and all those things. But for now, we're going to take a step back and talk about glycogenesis and glycogenolysis. So what is glycogen? Glycogen, remember, is when we take all these glucose and it's all bundled up together, it's all branched together. So glycogenesis is when we produce glycogen using 2 big enzymes. One is glycogen synthase and another is a branching enzyme. And you'll learn a lot more about all these different enzymes that are involved. But for now, these are the 2 big enzymes that are occurring. This is mainly occurring in the liver and in the muscle cells. Glycogen is mainly stored in the liver. So you should think about the liver as this place where a lot of glycogen is getting stored, a lot of glucose is there, and liver is really responsible for all those things. Your liver is a really big organ. Glycogen synthase is creating alpha-1,4 glycosidic bonds between glucose. And if you don't know what that is, then revisit I think it's the biology video on that. And then there's branching enzyme, which is creating branches doing branching with the alpha-1,6 glycosidic bonds. Then we have glycogenolysis, which is the breakdown of glycogen into glucose using glycogen phosphorylase and debranching enzymes. Glycogen phosphorylase is removing a single glucose 1-phosphate molecule by breaking the alpha-1,4 glycosidic bond. In the liver, it's activated by glucagon to prevent low blood sugar. In exercising skeletal muscle, it's activated by epinephrine and AMP, and that allows for glucose to be provided to your muscle, which is like exerting strength to move objects. Debranching enzyme is moving a block of oligoglucose from the branch and connecting it to the chain using an alpha-1,4 glycosidic bond. It's removing the branch point, which is connected via alpha-1,6, releasing a free glucose molecule. This will make sense if you look at a diagram, which we can do over here. All right, this is not the diagram. Okay, this is the diagram. So this is the process of, okay, I guess it might not be in this document, so I'll show you a Google image. So this is about as simple of a diagram as I can find about what is going on in glycogenesis. So remember, glycogenesis is the process of taking glucose molecules, which are these individual sugars, and then we stack them up and build them to each other so that we can form glycogen. Glycogen is our main storage molecule in the liver, and so when your body needs fuel, then the liver is going to release and break down glycogen into glucose molecules that the rest of your organs can now use and convert into ATP. So basically what's happening in glycogenesis is the production of glycogen. And so you have a glucose, the glucose with the help of hexokinase becomes glucose 6-phosphate. So remember, that's also the first step of the process of glycolysis, right? Then we do a mutase and we change this glucose 6-phosphate and we put it onto the 1 position. So now we have glucose 1-phosphate. Now we basically we're replacing that phosphate with a UDP. And so you can see here we're putting in a UTP, we're releasing 2 phosphates, and what's happening is we have UDP glucose. Now UDP is getting released, and through glycogen synthase we're going to convert this glucose into with the help of branching enzymes. And so there's a couple of steps that's happening here, and eventually we form glycogen. And so it's important to know that there's glucose. With glycogen, you have a lot of alpha-1,4 bonds that are connecting these glucose, and every once in a while you'll have alpha-1,6 bonds that are branching off, and that's kind of what's happening with glycogen. Now let's talk about glycogenolysis. So this is the process by which you have a lot of glycogen and you want to break it down into individual sugar molecules or individual glucose. So what happens is you have glycogen, you do glycogen phosphorylase, this becomes individual molecules of glucose 1-phosphate. Now obviously there's a couple of steps that are going on in between, but this is the major step. Now you have a glucose 1-phosphate, so you have to do a mutase that'll switch the 1 position to a 6 position. So you have glucose 6-phosphate. Now you can just convert that into glucose. This can happen in the liver and kidney with the help of glucose 6-phosphatase. Or this glucose 6-phosphate, now you can just make it participate in glycolysis, and it breaks down into pyruvate and then acetyl-CoA that gives off carbon dioxide, and this participates in that Krebs cycle that we were talking about. It creates ATP, and the electrons participate in the electron transport chain. Now, all the diagrams of gluconeogenesis are a little bit more complicated, but fortunately you don't really need to know that much for the purposes of the MCAT. All you kind of need to know is that if you have a lot of lactate, or you have a lot of pyruvate, or you have a lot of like 2-phosphoglycerate, right? These are all things that might accumulate. And so the cell might not want to produce any more ATP. It might want to actually regenerate glucose molecules for other purposes. So what can happen is gluconeogenesis. And if you think about this, glucose, neo is new, and genesis is creating. So you're creating new glucose molecules, and so you're reversing this process. And by putting in ATP molecules, you can build back up glucose. This is an anabolic process, and you can see in the liver this happens all the time with you have a lot of lactate, and you can actually you can bring back glucose. And this is a fortunate thing that our liver is able to do, because otherwise if you have too much lactate that's building up, then you might have lactic acidosis or some other bad thing in our body. And so instead you can use gluconeogenesis, and a variety of compounds can undergo gluconeogenesis in order to produce glucose. So over here, that's what I was talking about, gluconeogenesis. The two important enzymes that the sheet wants to talk about is the pyruvate carboxylase and fructose 1,6-bisphosphatase. So you know fructose 1,6-bisphosphatase already, because you know that fructose 1,6-bisphosphate can be converted to fructose 6-phosphate and bypass that phosphofructokinase 1. This is the rate-limiting step of gluconeogenesis, so that's kind of important to know. And then it's activated by ATP and glucagon. It's inhibited by AMP and insulin. Why? That should make sense to you. So insulin should inhibit this process. If you have a lot of insulin, that means you have a lot of blood sugar. That means you don't want to undergo the process of gluconeogenesis. AMP means you're energy deprived, and so you want to make glucose go into ATP molecules. You don't want to go the reverse way. ATP, just reverse that logic. And then glucagon basically means that you don't have a lot of blood sugar. Your body wants to produce more sugars. And so if you have glucagon, that means that you want to produce ATP. And so that's going to activate this enzyme and create more sugars. Pyruvate carboxylase is kind of a similar thing to know. This is a really interesting enzyme. It converts pyruvate into oxaloacetate. And then, so remember, pyruvate is that the end product of glycolysis. So pyruvate is formed by glycolysis, and then oxaloacetate is one of the last substrates that's participating in the Krebs cycle, right? And so usually pyruvate has to become acetyl-CoA, and then the acetyl-CoA has to undergo all these steps in order to become oxaloacetate. But pyruvate carboxylase, what it does is that you have this pyruvate over here, and it can just directly produce oxaloacetate over here in that Krebs cycle. And so what's happening is that it's converted from 2-pep by pep-CK, and then together those two enzymes, they bypass pyruvate kinase. And what happens is that pyruvate carboxylase is then activated by acetyl-CoA, and pep-CK is activated by glucagon and cortisol. Again, I think this is going to make more sense if I show you a picture, so that's what I'm going to do. So this is a pretty good image of talking about what I was saying. So pyruvate, instead of becoming acetyl-CoA, can actually bypass that step through pyruvate carboxylase and form oxaloacetate, which is one of the substrates in the Krebs cycle. And oxaloacetate can then become aspartate that can participate in the urea cycle and produce ammonium, or aspartate can go into gluconeogenesis and we can form new glucose molecules. And so that's what that means basically, is how does pyruvate participate in, it's a different way that pyruvate can participate in gluconeogenesis. Okay, now let's talk about the pentose phosphate pathway. So the pentose phosphate pathway is basically, it's also known as the HMP shunt. And this is a really interesting pathway that's a little bit different from what we've talked about, but it is important because it involves sugars for biosynthesis and it involves NADPH. So basically, let me show you a pathway diagram and then I'll explain what's going on. So I think this is a pretty good diagram that doesn't simplify this pathway too much. So what's happening is you have these hexose sugars. This can be glucose, it can be ribose, sorry, not ribose, it can be glucose, it can be fructose, right? So basically the hexose sugar is going to participate, it's going to form glucose 6-phosphate. Then we'll undergo glucose 6-phosphate dehydrogenase. You'll convert an NADP plus to an NADPH. Remember, NADPH is one of those things that's important for oxidizing things, for reducing things. And so this is pretty important, for example, in places where you might have a lot of free radicals. And then glucose 6-phosphate will form 6-phosphogluconate. That's not too hard to remember. It'll undergo this step, which is not too important for you. And then it'll undergo this last step, which is forming ribulose 5-phosphate. Now this is all oxidative phase. This is the oxidative phase. It's producing these reducing compounds, NADPH, right? 2 of them, and you took basically a glucose or a fructose and you formed a ribulose 5-phosphate. Now, what are we going to do with this ribulose 5-phosphate? Now we're going to undergo this process, which is the non-oxidative phase, and this is the part where you form these interesting, important compounds, namely something like ribose 5-phosphate. That's kind of the important one that they want you to know about. So this ribulose 5-phosphate can undergo this isomerase process to form ribose 5-phosphate. This can participate in DNA synthesis, RNA synthesis, all those types of things. There's also this transketolase enzyme. You can form different types of these sugars, and so the very important one that they'll want you to know about is that it can form ribose 5-phosphate. The other one that they might want you to know is that in the non-oxidative phase, you might form fructose 6-phosphate. So here, let's go through what's going on. So you have the HMP shunt. It's in the cytoplasm of most cells. You have glucose 6-phosphate that enters that pathway, and the product is 2 NADPH, sugars that can undergo biosynthesis, and then glycolysis intermediates that can undergo glycolysis or gluconeogenesis. The rate-limiting enzyme here, this is important to know. Generally, rate-limiting enzymes are pretty important for you to know, is glucose 6-phosphate dehydrogenase, which is the enzyme that's acting on the glucose 6-phosphate. And this is an enzyme that's activated by NADP plus and insulin and inhibited by NADPH. Generally, you should always know by intuition what enzymes are activated and inhibited by what. So it should make sense. Insulin means you have a lot of blood sugar. If that's true, then you want to use glucose and make it do interesting things that are not just like being glucose. So that could be, participating in the HMP shunt. NADP plus, if you have a lot of that, that means that you don't have a lot of NADPH. And so you want to undergo this process, which is converting NADP plus into NADPH. And if you have a lot of NADPH, then you don't want to undergo this process because that's going to convert even more NADP plus to NADPH. All right, now we have other monosaccharides. This can be galactose and fructose, and in med school, you're going to learn a lot about different childhood diseases that can happen in babies who are not able to consume galactose or fructose. But galactose is the big one in milk. It comes from lactose in milk, and it's trapped in the cell by galactokinase. This is the enzyme that helps to break it down, and it's converted into 1-phosphate via, basically, it's always hard to remember what this enzyme is, but it's galactose 1-phosphate uridyl transferase, G1PU, and an epimerase. So just remember that there's this uridyl transferase, and you'll remember this enzyme. And then there's fructose. Fructose comes from honey, fruit, sucrose. It's trapped in the cell by fructokinase and then cleaved by aldolase B to form glyceraldehyde and DHAP. Remember, these two compounds, they were important in our glycolysis pathway. So fructose, remember the two enzymes are fructokinase and aldolase B. Galactose, remember the two enzymes is galactokinase and GLP uridyl transferase. Okay, let's keep going. So now we have aerobic respiration. This stuff should be a little bit easier because this stuff was covered in your biology class, your biochemistry class, all those things. They love to test this in school and in the MCAT. So we'll spend a little bit of time, again, going through diagrams and making sure you understand this. So if you remember from our glycolysis pathway, we went through glycolysis. We had a glucose molecule that split up, and then we ended up with pyruvate. The pyruvate then became acetyl-CoA, right? And so now we're at acetyl-CoA, and this acetyl-CoA is now ready to participate in the Krebs cycle, which is everybody's favorite cycle. And now we need to learn, how does this acetyl-CoA participate in the Krebs cycle? So acetyl-CoA contains this CoA thing, which is a high ester thioester bond. It can be used to drive other reactions when hydrolysis occurs. So just know that this contains a lot of energy here. Acetyl-CoA formation, it can be from fatty acids. It can happen, that can form fatty acyl-CoA, which I'm sure you've heard of. Acyl is a fatty acid group that can be transferred to carnitine. Then that becomes acyl-carnitine. Acyl-carnitine is really important for transversing the inner membrane of the mitochondria. The acyl-CoA, the acyl group can be transferred to a mitochondrial CoA to reform the fatty acyl-CoA, which can undergo beta oxidation to form acetyl-CoA. I wouldn't worry about this part too much. Just know that carnitine is really important for helping shuttle this acetyl or this acyl-CoA thing. That's kind of all you really need to know for the MCAT purposes. You'll need to know more about this in med school. And then can be formed from the carbon skeletals of ketogenic amino acids, ketone bodies, and alcohol. So I would know that, you know, if you heard like anything about the keto diet and how everybody's in love with it for a period of time, something that they used to say is that you can form acetyl-CoA from ketogenic amino acids, ketone bodies, alcohol, all those things. Okay. Next is pyruvate dehydrogenase, which we've talked about already, but pyruvate dehydrogenase, it oxidizes pyruvate, creating CO2. It requires thiamine, pyrophosphate, and magnesium. Then we have these enzymes, which I won't focus on too much because I've never seen any question asked about either of these enzymes, but just for completeness, dihydrolipoyl transacetylase. This oxidizes the remaining 2 carbon molecules using lipoic acid and transfers the resulting acetyl group to CoA, forming acetyl-CoA. And then dihydrolipoyl dehydrogenase, it uses FAD to reoxidize lipoic acid, forming FADH2. So basically you need to know, or you don't need to know this, but basically what this is saying is dihydrolipoyl dehydrogenase, it uses FAD to form FADH2. And dihydrolipoyl transacetylase is, as it sounds, doing the transacetylase thing, which is transferring acetyl-CoA to CoA, forming acetyl-CoA. It's transferring the acetyl group to CoA, forming the acetyl-CoA. Okay, next let's talk about pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphorylase. These are interesting enzymes because they're basically enzymes of an enzyme. Kinase is something that phosphorylates things, and phosphatase is something that dephosphorylates things. The kinase phosphorylates PDH, pyruvate dehydrogenase, when ATP or acetyl-CoA levels are high. So it turns it off. That should make sense. So you're phosphorylating PDH, and that's going to turn off PDH. And remember, PDH is something that helps glycolysis continue into the Krebs cycle. And when ATP and acetyl-CoA levels are high, that means that you already have a lot of the end product, and so you want to decrease the amount of that process going through. What's always confusing to me is generally kinases help pathways move forward, and in this case, the pyruvate dehydrogenase kinase is turning off the enzyme, which is a little unusual, but that's just kind of how this one works. Then pyruvate dehydrogenase phosphatase, it's removing the phosphate group from pyruvate dehydrogenase, and it's turning it on, which means that it's helping the glycolysis proceed to the Krebs cycle. Okay, and then this is the carnitine thing I was talking about. Again, you're probably not going to get a question on this. You will get questions on this in the future in med school, but for this purpose, I would just know that basically there's this carnitine thing. That's a molecule that helps acyl groups cross the matrix, and so you can't actually cross with the CoA. And so what happens is this CoA becomes acyl-CoA, and this acyl-carnitine becomes carnitine. Now this carnitine is able to translocate to the other side of the membrane. The carnitine becomes an acyl-carnitine, and this acyl-CoA becomes a CoA. So what's happening is basically you're technically just shuttling CoAs across without actually shuttling that CoA across. So this is the mechanism for acetyl-CoA to enter the mitochondrial matrix. The mitochondrial matrix is where the acetyl-CoA can undergo beta oxidation to form acetyl-CoA. So if none of this made sense, just know that you're trying to get acetyl-CoA to enter the mitochondrial matrix, and you use carnitine to help you out. Okay, now we have oxidative phosphorylation. This is where we get to pretty interesting things, and we have reaction of citric acid cycle. So I'm going to continue to try to go in order, and we're going to go with the citric acid cycle or the Krebs cycle, which is everybody's favorite. And these enzymes are not going to make a lot of sense unless you look at a diagram, so we'll do just that. Unfortunately, this diagram is really beautiful in this review sheet, so we'll just go through this. And there's a lot of mnemonics that for some reason I never learned until I started med school, where we had a really good mnemonic, so I'm going to show you that one. So the mnemonic that I use to always remember these substrates, because you're going to forget it no matter what, is, and you can use this if you want, or you can try to find your own way of memorizing it, but what I use is citrate is Krebs starting substrate for making oxaloacetate. So you do have to remember that citrate is the first one and oxaloacetate is this last one. But if you can remember that, then that's how I remember it. So citrate is isocitrate. Citrate is Krebs alpha ketoglutarate starting succinyl-CoA substrate succinate for fumarate making malate oxaloacetate. And the nice thing about this is the words also kind of sound similar. So citrate is isocitrate, Krebs alpha ketoglutarate. That's the only one that you kind of have to stretch a little bit. Starting succinate, substrate succinate. So succinyl-CoA is starting, and then succinate is substrate, and you get the point. For fumarate, that's a really good one. So basically, we have these series of substrates that are going through the cycle. The reason why it's called Krebs cycle is because oxaloacetate reproduces citrate, and so this thing just keeps going in a cycle. And what you need is acetyl-CoA to enter the cycle. Acetyl-CoA is going to join with oxaloacetate to form citrate, and so that's how the cycle starts. So now let's talk about what's going on. You don't actually need to know what these structures look like. You might want to know like which one is a 6-carbon, which one is a 5-carbon, which one is a 4-carbon, but you will know that based on where the carbon dioxides are leaving. So let's just go through the steps. First step is citrate synthase. So you're forming citrate. How are you doing that? Well, you're taking the acetyl-CoA that we took, that's the acetyl-CoA, and we're combining oxaloacetate, which is the last step in the cycle, and you're combining them to form citrate. Citrate is a 6-carbon molecule. That should make sense because you have 2 carbons right here, and you have 4 carbons right here. Now the CoA is falling off here, so the CoA was just kind of this thing that was helping us proceed to this point, and then it's going to fall off. Now we have citrate, which is our starting substrate, remember? So citrate is isocitrate, the starting substrate. We have this enzyme that you don't need to remember, aconitase. Isocitrate now undergoes isocitrate dehydrogenase. This is a rate-limiting step, so I would know that. Isocitrate dehydrogenase, in this step, you're going to produce 1 molecule of NADH, and you're going to pop off one of the carbon dioxides. So now, this, remember, was a 6-carbon molecule. What do you think it'll become now? It's going to be a 5-carbon molecule. And count that, 1, 2, 3, 4, 5. Now you have alpha ketoglutarate. So remember, citrate is Krebs alpha ketoglutarate. Now alpha ketoglutarate is going to undergo this enzymatic process. I love this enzyme because it's such a nice name, alpha ketoglutarate dehydrogenase complex. Now you're going to release another molecule of NADH. You're going to release a CO2, and so basically you get the same things that are released over here as here. Now remember, this is a 5-carbon compound. We're releasing a CO2, so this should be a 4-carbon compound over here. So now you have succinyl-CoA. So remember, CoA is getting added again. That's what's happening over here. So this alpha ketoglutarate dehydrogenase complex is doing a lot. It's removing a carbon, it's reducing an NAD plus into NADH, and then it's also adding a CoA. Now we get succinyl-CoA. We need to now form an ATP, and we need to get rid of that CoA to form succinate. Remember, this is a 4-carbon compound with a CoA. We're going to remove a CoA. We're going to generate an ATP using succinyl-CoA synthase, and we'll get another 4-carbon compound that doesn't have the CoA. That's succinate. Now you get succinate dehydrogenase. Remember, dehydrogenase things are basically reducing something, so it's going to reduce FAD into FADH2, and then you're going to form fumarate. Fumarate is another 4-carbon compound. Fumarate undergoes an enzymatic process through fumarase for making malate. So remember, citrate is Krebs starting substrate for fumarate making malate. So fumarate becomes malate. This is through fumarase, and malate again is a 4-carbon compound. Now you go through malate dehydrogenase to form oxaloacetate. Oxaloacetate is the final substrate in the Krebs cycle, and this is another 4-carbon compound. Now, I think what's important for you to remember, or just try, this is really hard to remember. You're inevitably going to memorize it and then forget it the next day, or remember it for your exam and then forget it the next day. It's kind of the natural process of medicine. This is the one thing that everybody remembers and forgets after taking their exam. But basically, you want to try to remember, where are you producing these NADHs, where are you producing the CO2s. So we produce an NADH in this first enzyme from isocitrate to alpha-ketoglutarate. We form an NADH again from alpha-ketoglutarate to succinyl-CoA. You form an ATP from a succinyl-CoA to succinate. You form an FADH2 from succinate to fumarate. And then you're not producing anything here, but you do produce an NADH from over here, from malate to oxaloacetate. So I can remember first step, nothing's happening. Then second, third, and fourth step, and fifth step, there's a lot of things happening. So in this area, there's a lot of things being produced. I always remember that the last one in this area is FADH2. I remember that the first two is NADH over here, and then in the middle, you're not producing NADH, but you are producing ATP. And then this very last one is producing another NADH. So what's the main thing that Krebs cycle is helping us with? If it's just kind of a cycle, it's not producing any new compounds for us to take out, then why is this so important? Well, the reason that this is so helpful is because we're producing a lot of NADHs, and we're also producing an FADH2, right? And so these have a lot of electrons, and so that's why this cycle is so important, because it's helping us gather a lot of electrons from the citrate. And so this citrate has basically a lot of electrons that we're taking electrons out of this, and that's why we end up with oxaloacetate. Obviously, there's a lot of other things that's going on to produce oxaloacetate, but the main thing that we're getting out of this is we're oxidizing this compound a lot. All right, now we're going to move on to our final step, which is the electron transport chain, which is why did we try to gather all these electrons in the first place? Why did we go through all this effort? It's to make these electrons do work for us so that we can produce ATP. So we basically just covered all of that, which is a lot of information. And I would read through it on your own time, all of these enzymes, but basically we just covered what all of these enzymes do. And again, for biochemistry, diagrams is the #1 thing, which is going to help you memorize things, remember things, understand things. You're not going to get a lot out of just reading things, especially for biochemistry, and especially for these pathways, so I'd recommend and not just these sheets. This is something where I watch YouTube videos, animated YouTube videos, to try to understand like where things are going, what things are doing. And so just you're going to get a lot more from watching a 3-minute video on these pathways than you will from reading for 10 minutes about these enzymes. So that's what I would recommend for learning biochemistry again. Now let's move on again to the electron transport chain. Why did we gather all these electrons? It's to funnel them through this chain that takes place on the matrix-facing surface of the inner mitochondrial membrane. Basically, you have all these NADHs, you have also some FADH2. They're going to donate electrons to this chain, and those are going to be passed from one complex to the next complex. And you can imagine that as the electron is moving, it's generating some reduction potential, and it's going to finally, after it's getting passed on from complex to complex, it's going to reach oxygen, which is going to receive these electrons. Oxygen is going to get these electrons, and it's going to form H2O. So the complexes, again, are not that important for you to know, but I would know that these are all, these are like, basically you jump from one complex to the other complex. These electrons are passing through in the chain. That's the major thing you should know. You can read through these complexes, but again, it's not, some things in this document are included for completeness sake, but this is something that I wouldn't necessarily bore you about. You can read it just once and try to understand like what's going on in each complex. Then you have NADH cannot cross that inner mitochondrial membrane. So it's staying on the outside, and there has to be a shuttle mechanism to transfer electrons into the mitochondrial matrix that's allowing it to be used. So these are basically the two shuttles that are allowing NADH to cross the inner mitochondrial membrane. The first is the glycerol 3-phosphate shuttle, and this is basically electrons are transferred from NADH to DHAP, forming glycerol 3-phosphate. These electrons can then be transferred to mitochondrial FAD, which is forming FADH2. So you are losing some energy here from going from NADH electrons to FADH2 electrons, and then the other one is malate aspartate. So electrons are transferred from NADH to oxaloacetate. This forms malate. So remember, both of these are part of the Krebs cycle. And then malate can cross that inner mitochondrial membrane, and then it transfers the electrons back to mitochondrial NAD+, forming NADH. So those are the two ways. One way is that you're keeping the electrons with NADH. The other one is you're transferring NAD electrons to FAD, essentially. Okay, so we went through all of that. I just want to show you a diagram to understand the electron transport chain, because this is kind of another pretty high yield one. All right, so this is oxidative phosphorylation, also known as the electron transport chain, also known as chemiosmosis. In eukaryotes, it occurs in the mitochondria, like we've been talking about. Prokaryotes don't have mitochondria, so it's referred to as, it's occurring in the cell membrane. So this is the inner mitochondrial membrane over here. These are the complexes, and electrons are passing from one complex to another complex, and they're hopping through, and as they pass on, they're creating a potential gradient. They're allowing protons to move from inside to outside. And then one thing I want to clarify is that this is the inside, this is the outside of the inner mitochondrial membrane. So this is the inner mitochondrial membrane, this is the intermembrane space, this is the inside. And so protons are moving to the intermembrane space. It's now accumulating over here, so we get a positive charge over here. And because protons are leaving here, this gets a negative charge. And as these electrons are hopping from complex 1 to complex 2 to complex 3, it also hops to the Q complex. So you have complex 1, which is NADH dehydrogenase. This is where the NADH electrons leave its electrons, so this is where they accumulate. Complex 2 is succinate dehydrogenase. This is where the FADH2 electrons leave their electrons. So you can see that the NADH electrons are actually leaving at an earlier complex, and so that's why these electrons are more valuable for producing energy. FADH2 leaves it at a later complex, complex 2, and so that's why these electrons are a little bit less valuable in terms of producing ATP. Now they keep going. Complex 3 is cytochrome reductase, and then complex 4 is cytochrome oxidase. Finally gets to complex 4, and then it combines with oxygen, 2 protons, to form water. Right? So that's where, when you remember this whole reaction of glucose forming ATP, you'll remember that there's oxygen that's being taken up, and you remember that water is being produced. That's where this is coming from. So this is, again, an important reaction. It requires oxygen for all of this to occur. If you don't have oxygen, then this cannot occur. And protons are all accumulating in this space over here. So now you have all these protons that are forming a positive charge here, and they all want to come back. They can actually come back through this ATP synthase. The ATP synthase will allow these protons to come back into this inside, and this is what's referred to as chemoosmosis. And basically what's happening is every proton is returning back, it's twisting this thing. And this ATP synthase, as it's twisting, is actually taking an ADP and a phosphate, and it's combining them. That's what that twisting motion is doing, and that's how you result with ATP over here. And that's basically the end of our whole process of taking glucose and forming water and ATP and taking up oxygen. That's how all of that is occurring. That's why this is so valuable. So why do you think our cells go through all that effort? Why can't they just like burn up a glucose molecule and try to generate energy from that? It's because this is a really controlled thing that's taking a lot of steps. So you have this glucose, which is all the way up here, and you want to continually extract energy from it and take as much energy from this glucose molecule as possible. So if you just burn this glucose molecule, you might generate some amount of energy, but in this controlled fashion, the cell is able to convert it into ATP that the cell is able to use better. Glycolysis produces 2 NAD and 2 ATP. That's forming 7 ATP. pyruvate dehydrogenase complex, that's forming one NAD for every one pyruvate, so essentially 2 NAD, which is another 5 ATP. Citric acid cycle, it's forming 6 NAD, 2 FAD, 2 GTP, it's forming 20 ATP. And this is all of these NADs are contributing to the electron transport chain. So ultimately, one glucose is forming about 32 ATP. I will say that don't memorize these numbers as absolute, because in the literature, there's some controversy about exactly how much NADH and how much FADH contribute to each ATP. But essentially, this is kind of how I learned it as well, is that each NADH contributes to 2.5 ATP. Each FADH, 2, contributes to 1.5 ATP. And so basically one glucose contributes to somewhere between 30 and 34 ATP, somewhere around that number. And so, yeah, so that's how that works. That's why our cells go through all this effort in order to produce a lot of ATP. All right, so we just did a great job. We covered some of the most high yield and important things in the biochemistry section. Usually when people ask me to tutor them on biochemistry, they're not actually talking about all these other pages. They were talking about page 9 and 10, which are the really big pages of all the pathways, all the enzymes, all the tough things to memorize. And again, I'm just going to repeat that this video is meant to be comprehensive, but to memorize all these things, instead of reading back through these sheets, I would recommend going to other YouTube videos that show animations, that kind of show diagrams, and just trying to absorb this information. Go to Google Images and look at more diagrams that explain all these things. The really nice thing about science and biochemistry and the MCAT is that, unlike more theoretical things like math and physics, these things are things that you can look at pictures of and understand and memorize. And so that's how I would recommend that if you're reviewing biochemistry, you focus on these 2 pages and you look at as many diagrams and pictures and videos as possible. Okay, now we have lipid and amino acid metabolism. So how do lipids and amino acids contribute to this whole picture of how we are using the food that we digest and converting it into energy? So the first thing is mechanical versus chemical digestion. Mechanical is the mouth and the stomach. Stomach, there's acids that are breaking down things. The mouth, you're chewing things up. Chemical is you have lipids that are being broken down in the small intestine. You have bile, you have pancreatic enzymes like lipase, you have cholesterol esterases, you have all these things chemically that are breaking down the food that you're eating. Then you have emulsification, which we've talked about already, but the idea is that you're entering the duodenum, that's the thing after the stomach, and you have emulsification, which is that you're mixing 2 things that normally don't mix together, like fat and water. So another example is oil and vinegar salad dressing. And basically you're increasing the surface area of the lipid, which is allowing more enzymatic interaction and processing, and it's helped by bile salts. And that's why bile salts are so valuable, because it's helping you digest all these lipids. My cells, remember we've talked about that. That's the thing that's involved in things like soap. You have water-soluble spheres with a lipid-soluble interior. So because of this, the whole thing is can go into water and mix into water, even though the majority of the interior is actually lipid-soluble. Then you have short versus long fatty acids. Short chain are absorbed across the intestine into the blood, and long chain fatty acids are absorbed as micelles and assembled into chylomicrons for release into the lymphatic system. Okay, now, let's talk about this. Not super high yield, but this is the lumen, this is the mucosal cell. The mucosal cell, you will have chylomicrons that are entering into the lymphatic system. And in the lumen, you have all these triacylglycerides from the things that you're eating that's getting broken down by lipases into fatty acids and monoacylglycerols. Those can combine after they cross this membrane and form triacylglycerols. They can combine with other lipids and proteins. They can form chylomicrons, which again participate in that lymphatic system. Okay, now we have lipid mobilization and transport. Lipid mobilization is lipids are mobilized from adipocytes by hormone-sensitive lipases. Lipases are mobilized by lipoprotein lipases. So this is basically just how lipids are moving around. They can be mobilized from lipoproteins or they can mobilize from adipocytes. This is how they're getting chopped out and how they're moving from different places. Chylomicrons, they transport dietary triacylglycerols, cholesterol esterases. You'll have to know this a lot more in detail for your first board exam in med school, but for the purposes of MCAT, you kind of just need to know that they're important for the lymphatic system, and they're kind of the first thing that's in your gut that's helping to absorb all these triacylglycerols and cholesterol esterases that you're eating in your intestine and transfers it to tissue. Then you have lipoproteins. Lipoproteins can be VLDL, intermediate, low density, high density. I always remember H for healthy, so HDL, high density lipoproteins or high density lipids, those are good. And LDL is bad. And so that's kind of like a dietary standpoint. If you're a doctor and you're reading like all the results of somebody's lab studies and you see HDL is like above some number and LDL is below some number, then you'll tell them like, oh, that's great because LDL is bad and HDL is good. And so HDL is kind of helpful for scavenging. It moves cholesterol into the liver that can exit the body. LDL is kind of bad because it's moving cholesterol into the tissue. This is a kind of a gross simplification, but generally that's kind of what you need to know. I wouldn't worry too much about intermediate and very low densities. But just generally, I would know that these are lipoproteins are how these lipids are transporting. And then you have apolipoprotein, which is the protein component of the lipoprotein. So lipoproteins are kind of looking like these circles that carry like cholesterol and carry different lipids across from tissue to liver to other places. And then apolipoproteins is in this membrane, you have some protein in it. That's what apolipoproteins are. They're also receptor molecules that control interactions between, and then the spread cuts off. So I'm not sure what he was trying to write here, but they're basically protein components of lipoproteins. Okay, now we have cholesterol metabolism. Cholesterol can be obtained from dietary sources or through de novo synthesis. So the liver can synthesize cholesterol. And the important enzyme to remember, the really big one that a lot of medications work on, is HMG-CoA reductase. That synthesizes mevalonate, and that's the rate limiting step of cholesterol synthesis. So this is the really big one that a lot of enzymes or a lot of medications target to try to stop this from working, because this is what helps synthesize cholesterol. Then we have LCAT and CETP. So LCAT, LCAT, this catalyzes the formation of cholesterol esterase for transport with HDL. And then you have CETP, which is catalyzing the transition from LDL to IDL by transferring cholesterol esterases from HDL. I will tell you that all of this stuff is kind of hard to remember and get in your head. I would try to read this at least once, make sure you kind of understand it, but I never really got too many questions on lipid mobilization, transport, cholesterol metabolism. And looking at images is going to help you a lot more. And looking at an animated video is going to help you really well for these stuff. So if you feel uncomfortable with this subject material, even though it's not as high yield, I would recommend watching an animated video to try to understand all these topics. And within 10 minutes, I think you'll know everything you need to know for the MCAT regarding these two. Okay, now we have fatty acids and triacylglycerols. You already know a lot about fatty acids and a lot about triacylglycerols, so I won't belabor the point, but I think understanding kind of the biochemistry behind them is important. All right, so next let's talk about the fatty acids and triacylglycerols. Fatty acids are carboxylic acids with a long chain. They can be saturated or unsaturated. Unsaturated means they have one or more double bonds, and saturated means they have no double bonds. So when people tell you don't eat saturated fats, what they're telling you is don't eat these types of don't eat fats with a lot of these saturated fatty acids, because basically, since they don't have any double bonds, they don't have any kinks in the chain, and so they're straight. And what that means is that you can pack a lot of these fatty acids together, and so they're really packed with a lot of calories, and that's why people say don't have a lot of saturated fats. Fatty acid synthesis is synthesized in cytoplasm from acetyl-CoA, and it's transported out of the mitochondria. So I remember that carnitine is one of the really important things for transporting acetyl-CoA, and I remember that inside the mitochondrial matrix, that's where fatty acids are broken down, and outside the mitochondria, where the cytoplasm is, that's where acetyl-CoA is being used to build up fatty acids. So in the cytoplasm, there's building of fatty acids, and inside the mitochondria, there's fatty acids getting broken down. Happens in 5 steps. Fortunately, you don't really need to know these in a lot of detail, but there's activation, bond formation, reduction, dehydration, and then finally second reduction. Arachidonate is also important. It's a precursor to those icosanoid signaling molecules. So these can be prostaglandins, prostacyclins, thromboxane. They're also precursors to leukotrienes. This whole pathway is really important for a lot of medications that people use, including things like aspirin, a lot of NSAIDs that you might take for pain. They kind of block a lot of the synthesis of leukotrienes and all these things, but for your purposes, it's just important to know that arachidonate is a precursor to those signaling molecules like prostaglandins, prostacyclins, thromboxanes, and those play a really big role in traveling through your bloodstream and doing a lot of signaling. Now you have fatty acid oxidation, and fatty acid oxidation is something that occurs in the mitochondria, like I was telling you before. It happens with a carnitine shuttle. I remember carnitine shuttle for carnage, and that helps you oxidize fatty acids, and oxidation of fatty acids is basically breaking them down. Beta oxidation uses cycles of oxidation, hydration, oxidation, dialysis, cleavage. Again, not too important that you know these steps. More important that you know like everything we talked about in the first 2 pages, or the 2 pages previous to this. But the fatty acid is shortened by 2 carbon atoms at a time. So let's say you have a 16 carbon fatty acid chain. You'll keep like whittling it down by 2 carbons. So you'll have 14 carbons, then 12 carbons, then 10 carbons, etc. And the fatty acid is shortened by those 2 carbon atoms. And then you have FADH2, NADH, acetyl-CoA that are generated from this breaking down. And this is assuming that it's even. If there's an odd number, then you might get a carbon dioxide that's released. Yeah, so this is just showing what the fatty acid looks like. You have like these double bonds. So if you remember what I was talking about before, because there are these double bonds, that means that this is unsaturated. There's one or more double bonds. And the carboxylic acid end, this end is called the alpha end, and this end, I think this is omega. That's the omega end. And then these are different types of fatty acids that they're just showing you examples of. All right, now we have ketone bodies. This is what everybody was obsessed with during COVID, is ketogenesis, ketosis, the keto diet. And this is kind of some of where the biochemistry comes from. Ketogenesis is the production of ketone bodies. That happens when you have a lot of excess acetyl-CoA in the liver during a prolonged starvation state. So when we have people in the emergency room who show up and they look just kind of cachectic and they're very malnourished, and it seems like they're kind of unkempt, something that they look at is the urine, and they look at ketones in the urine, and they also look at the glucose in the blood. And something that they might have is ketones in the urine, and they have normal glucose or low glucose, and that kind of tells you that they have ketogenesis. They have something called starvation ketosis, which is where they are generating ketone bodies because they're not eating enough, and so the body is now relying on ketone bodies instead of glucose for energy. Ketolysis, so whenever you see lysis, that means breaking down. Ketolysis regenerates acetyl-CoA from the ketone bodies. And then you have energy source. The brain is a big reliant on ketone bodies during starvation, so your brain can use those ketone bodies during prolonged starvation. And so that's why, I don't know if it's true or not, but if you watch YouTube videos of people who've tried the keto diet, a lot of them say that they're a lot sharper, and they, like, again, I don't know if this is placebo or if it's true, but a lot of people feel like because the brain is using ketone bodies, they are feeling more efficient, and they're just sharper. Okay, now let's talk about protein catabolism. Again, catabolism is breaking things down. Anabolism is building things up. Think about like anabolic steroids. People in bodybuilding, they're building their muscles up. Catabolism is breaking things down. Protein digestion occurs in the small intestine. So that should make sense. Right after your stomach, you have the small intestine. And that's where a lot of the digestion of proteins is happening, especially with the help of enzymes from the pancreas. Catabolism of cellular proteins occurs only under conditions of starvation. Amino acids are released from proteins, usually lose their amino group through deamination, and the remaining carbon skeleton can be used for energy. So you have 2 types of amino acids. I would know this for the MCAT, but it's okay to forget this right after because it's very hard to remember these things. But glucogenic amino acids are those that can be converted into glucose through that process, and you should remember what that process is, and it's gluconeogenesis. And all amino acids except for leucine and lysine are glucogenic. Ketogenic means it can be converted to acetyl-CoA and to ketone bodies. And leucine and lysine are the only two that are ketogenic. So remember, leucine, lysine, those are ketogenic. All others are glucogenic. And basically, this is saying that they're the only amino acids that are solely ketogenic. So there are amino acids that are both glucogenic and ketogenic. But leucine and lysine are the only two that are only ketogenic. They're not glucogenic. So just pay attention to that. Okay. I guess I can go over this. If I were to tell you not to memorize something, don't memorize this, as this is too much information and they're not going to test you on this. But just so that you're aware of what this is talking about, this is kind of how different compounds in the Krebs cycle can, how like you don't have to enter the Krebs cycle through pyruvate. You can enter the Krebs cycle through a variety of amino acids. So like asparagine, aspartate, those are amino acids that can enter through and become oxaloacetate. Arginine, glutamate, glutamine, histidine, proline, those can enter in through and become alpha-ketoglutarate. Leucine, lysine, phenylalanine, tryptophan, tyrosine, those can become acetyl-CoA, which then become acetyl-CoA. And remember, leucine, lysine, those are the ketogenic ones, and they're obviously eventually going to become acetyl-CoA, so that should make sense. Okay, so that's basically what that's talking about. Again, you don't need to memorize this, but that's a helpful diagram to understand what's going on, how your body is able to process a variety of things. All right, let's keep going. So now we have thermodynamics and bioenergetics. And so basically what this is talking about is a lot of the things that we've reviewed in the chemistry section. So I would recommend going through that if this stuff doesn't make sense. But you have open systems, closed systems, you have entropy, changes in free energy. So the easiest one to remember is entropy. This is how dispersed is energy, how disorganized is it. You can think of your room. If it's very disorganized, there's multiple states that it can be in if it's very disorganized. If it's perfectly clean, like all the books are in the shelves and all the clothes are on their hangers, there's only like one way that can happen, or a few ways that can happen. So entropy is how much is the energy disorganized, is it dispersed. The universe is always progressing towards a more entropic state with more entropy. Changes in free energy is calculated by standard free energy Gibbs energy, and it's delta G. And energy change in with this, you see this, I think it's standard conditions. I'm forgetting, it's been a while since I've been in chemistry class, but I'm forgetting what that dot, how you pronounce it, but basically that means that you're in standard conditions, 1 molar concentration, 1 atm, 25 degrees Celsius. And there's modified standard states. That means that you're in physiological conditions. Physiological conditions basically what your body's conditions are, and in your body generally the concentration of protons is 10 to the negative 7 molar. So pH in your body is 7. Blood pH is usually between 7.35, 7.45. Open systems means matter and energy can be exchanged with the environment, and then closed systems mean that only energy can be changed with the environment, but the matter stays within the environment. So think like a box or a cylinder. You're not like exchanging matter, but you're able to exchange heat. And enthalpy refers to heat exchange or changes in internal energy. Okay. Role of ATP. So we've talked all about glycolysis and production of ATP through the electron transport chain. What is ATP? ATP is a mid-level energy molecule. It contains these high energy phosphate bonds, specifically between the second and third phosphate, and it can be stabilized by hydrolysis. So remember, when you're going from a higher energy compound to a lower energy compound, the lower energy compound is more stable. When you're changing that transition, you're able to release energy. And so that's what ATP is useful for, is that by breaking that second and third phosphate bond, you're releasing energy, and that energy is used to do work within the cell that's helping your body to accomplish goals. So ATP is an energy source. Generally, ATP is 30 kilojoules per mole. Phosphoryl group transfer, so ATP can also not only hydrolysis and coupled to unfavorable reactions, but it can also donate its phosphate group. So in glycolysis, we talked about different reaction enzymatic steps where you're just donating the phosphate group to glucose, for example, to form glucose 6-phosphate. So it not only is useful for providing energy, but it can also transfer those phosphate groups. Okay, now we have biological oxidation and reduction. Biological oxidation and reduction can be broken down into component half reactions. Half reactions provide useful information about stoichiometry and thermodynamics. So there's flavoproteins and there's high energy electron carriers. So high electron carriers are the things that we were talking about, like NADH, FADH2, and then like cytochromes, those electron transport chain proteins, right? These are high energy electron carriers. They carry electrons, and then they can give those electrons at another place, and by transferring that the energy through the transfer of electrons, they're allowing work to be done. There's also flavoproteins like FAD, FMN, right? That's kind of a similar thing. They're a subclass of electron carriers, and they're derived from riboflavin, which is vitamin B2. And then I guess the other helpful tie-in of a possible way that they'll test this is that these are all half reactions. So when you're donating electrons from NADH to a compound, right, one thing is getting reduced and one thing is getting oxidized, and that's where you can have some tie-in from chemistry and physics. So that's how they might like try to integrate that, but usually they won't be that mean about it. Metabolic states, you can have equilibrium. Equilibrium is an undesirable state for most biochemical reactions because you need to be able to harness your free energy to survive. And so homeostasis is a good place to be. Your body is constantly trying to maintain homeostasis, like trying to maintain blood pH at a particular place, trying to make sure your respiratory rate is a specific thing, trying to make sure your blood pressure is a specific thing. But equilibrium is kind of like, are all the electron gradients completely fine? Is there like a gradient across the cell membrane? You don't always want that, right? Because you want yourself to be able to harness free energy and survive. Postprandial state, so postprandial means you just ate. Post-absorptive state means like you ate and you digested the food. Postprandial means you just ate, you're well fed, your insulin is up as long as you're like not a type 1 diabetic, and you're anabolic, so you're trying to you're trying to break down glucose, things like that. Sorry, you're not trying to break down glucose, you're trying to build up something like glucagon. And then post-absorptive, this is like you're fasting, but you had eaten already, your insulin is down, your glucagon is up, catecholamine is up, and you're transitioning to catabolism. So a helpful way to understand this is that postprandial, you just ate, there's insulin, there's anabolism, you're just trying to, you have a lot of glucose and you want to build that up into storage. Post-absorptive means you already ate, you kind of absorbed it, now you're fasting, and your insulin is down, your glucagon is up, your catecholamines are up, and now you're going to start to break down the sugars because your body feels the need to actually use those glucose molecules and make some ATP. Then you have prolonged fasting. So this is when you're like starving. Glucagon is really up, catecholamine is really up. A lot of tissues will rely on fatty acids. And then again, you'll have those ketone bodies that the brain can use. Okay, now you have integrative metabolism. Calorimetry, you've heard about it in your chem lab classes for sure. It measures metabolic rates. Respiratory quotient, that's basically how much are you actively consuming your fuel. So basically you calculate it by how much CO2 are you producing, by how much O2 are you consuming. And that's kind of a way that you're understanding how efficient and how much your body is using energy. Then you have regulatory hormones like ghrelin, orexin, and leptin. I always remember leptin keeps you lean, so that means that it's decreasing appetite. Orexin and ghrelin, I remember ghrelin means you're growling. If you're growling, if your tummy is growling, that means you're hungry. And so ghrelin increases when you have increased appetite, and then orexin also increases when you have increased appetite. Leptin keeps you lean, so that means decreased appetite. That's how I remember all of those things. So ghrelin and orexin, they kind of sound similar. Ghrelin and orexin, that happens when your stomach is growling, you're hungry, so you have increased appetite. Leptin keeps you lean, so that means that you're not feeling hungry, and that's when you have decreased appetite. BMI is just mass over height. A lot of doctors use that information, but also agree that it's kind of, there's a faulty system with BMI. because it doesn't tell you a lot of things like water weight and muscle mass and all these other things. Hormonal regulation of metabolism. So this is something I've been alluding to for a while. Insulin, glucagon, glucocorticoids, catecholamines, thyroid hormones. I would absolutely know this stuff, not just because it's high yield for the MCAT, but also because it's high yield for when you become a doctor. So insulin is something that is secreted by pancreatic beta cells. It's regulated by glucose, and decreased blood glucose means that you decrease blood glucose by increasing cellular uptake, increased rate of anabolic metabolism. Sorry, I'm just trying to read this. Give me 1 second. So yeah, what this is saying is basically when you have increased sugar in your blood, you're going to release more insulin from your pancreatic beta cells. Basically, that's going to be a stimulus for all the tissues in your body that we have a lot of glucose in our blood. We can take that glucose out of the blood, and then you can use it to build up things like glucagon storage for later when you're going to be in a fasting state or you're going to be starving, so that you can break down those glucagon for the production of glucose and ATP. So that's what that's saying. Then you have glucagon, which is basically the opposite. It's secreted by pancreatic alpha cells. It's stimulated by low glucose and high amino acid levels. And basically what that tells your body is that, especially your liver, is that we can increase gluconeogenesis and we can increase glycogenolysis. So we're going to break down glycogen and we're going to build up glucose because we're in a lower blood glucose state. Now we have glucocorticoids. Glucocorticoids is the way think about steroids, think about cortisol, think about those things. Basically, this is something that when your body's under stress, you're going to increase the blood glucose because all your tissues are under stress, and so they need to do more work. And so now the organs that have excess glucose, they're going to release it into the blood so that other tissues are able to take up this glucose and respond to whatever the stress is. Then there's catecholamines. Catecholamines include epinephrine, norepinephrine. There are think about like when your sympathetic nervous system is activated, you're running from a bear, or you're like in a fight or flight situation. You have this adrenaline rush. That's basically what catecholamines are helping you with. They promote glycogenolysis, right? You're going to break down your glycogen stores because you're in this immediate like high energy state and you need as much glucose as possible. Then you have thyroid hormones. Thyroid hormones are increasing basal metabolic rate. So when somebody has really high T4 levels or T3 levels, and you see like in their neck they have like this you call it like thyroid goiter, and you can feel it. Basically their thyroid is really hot and it's overactive and it's releasing T4 and T3, and what that is doing is it's like causing them like tremors, and it's making them like really active. They might have trouble sleeping, just tons of effects of hyperthyroidism, and like increased heat production, increased O2 consumption. Their like respiratory rate might be increased, and it's kind of the opposite with people who have hypothyroidism. They might have some like hair loss, their skin might be dry, they might like have a little bit cooler extremities, all those things. Thyroid hormones are tyrosine based, so I guess that can be like a biochemistry tie-in for this. Okay, I kind of talked about all this, but I would go through this diagram to make sure you understand it. Then we have liver, adipose tissue, skeletal muscle, cardiac muscle, and brain and nervous tissue. The one that they're going to test you the most on is the liver, so let's talk about that first. Liver is the most metabolically diverse tissue. People love the liver. It does a lot of really important things for us. Hepatocytes are like the cells of the liver, and they're responsible for maintenance of blood glucose level by glycogenolysis and gluconeogenesis in response to pancreatic hormone stimulation. The liver is also responsible not just for glucose control, but also for processing lipids, for cholesterol, for bile, for urea, for toxins. If somebody's an alcoholic, the liver is processing a lot of that alcohol and those toxins that's involved with the alcohol. So liver is very important metabolically for doing all these things, for maintaining blood glucose levels, for responding to pancreatic hormone stimulation, for processing lipids, for processing toxins, for processing urea, for processing bile, all those things. Adipose tissue. Adipose tissue is under the influence of insulin and releases them under the influence of epinephrine. So remember, epinephrine is like that fight or flight thing, right? It's a catecholamine, and it's telling you we want more glucose. And so your adipose tissue, which is your fat storage, it's going to tell it like break down and give us some glucose. It'll also, if you have a lot of insulin, if your blood sugar is really high, then you'll store those lipids in your fat tissue. Skeletal muscle, it depends on your activity level and fiber type. And so this should make sense. If you're resting muscle, if you're not doing anything with it, it's going to conserve carbs in glycogen stores and use free fatty acids from the bloodstream, right? It's not going to use its stores if it's resting muscle. But let's say you go to the gym, you're running a marathon, you're lifting some weights. Now it's going to use anaerobic metabolism. Anaerobic means without oxygen, and oxidative phosphorylation of glucose. So with oxygen, this is going to use direct phosphorylation from creatine phosphate. Creatine phosphate has that phosphate group, so there's going to be direct phosphorylation, and fatty acid oxidation. And it all depends on the fiber type. You'll hear about like type 1, type 2 fibers, exercise duration, so length of exercise, 1 second versus 1 hour. And fortunately, unless you're like an exercise physiologist or like a PM&R doctor, you don't need to know all the different like ways that muscle fibers will use all these different processes and like in which duration, of course, unless you become like a specific type of doctor that deals with that. Cardiac muscle is using fatty acid oxidation in both the well-fed and fasting states. So your heart is always beating, the cardiac muscle always needs to work, and so it'll use fatty oxidation no matter like what state you're in. If you're fasting, fortunately your heart is still beating. Then you have the brain and nervous tissue, and that's consuming glucose in all metabolic states except for prolonged fast. And again, in prolonged fast, like let's say like 48 hours you haven't eaten something, then your brain's fuel will start coming from those ketone bodies rather than glucose because your glucose is low. All right, that's everything for the biochemistry section. I hope that makes sense. I tried to include as many images as possible, include as many diagrams. Again, I'd really appreciate if you can hit subscribe. There's more content that's going to be coming out even past, even after I've reviewed all these milestone review sheets. I'm going to be coming out with more content. So I'd appreciate if you stay in touch and subscribe. And yeah, again, the best way to learn this is to watch videos, to look at diagrams, to look at different flow charts. It's going to help you memorize this stuff and remember it. This one is a real beast, very difficult to remember everything, but it's well worth the reward. There's a lot of biochemistry on the MCAT. So yeah, thanks so much for watching this video, and I'll see you in the next one. Bye, everybody.